geológicas bastante distintas, né? Mas tudo isso, então, vai gerar minerais e rochas carbonáticas que vão também ser depositadas em ambientes sedimentares muito distintos, né? Em ambiente tropical, água fria, né, com uma grande abundância de estromatólipos ou não, né? A gente pode ter ali diferentes ambientes, geralmente marinhos, com a presença de rochas carbonáticas. E aí eu vou ter, né, justamente por causa disso, eu posso encontrar diferentes faces carbonáticas, né? Estromatólitos belíssimos da Chapada Diamantina, ou microbialitos laminados, que às vezes podem se intercalar com alguns evaporitos, ou alguns carbonatos de retrabalhamento ali na região de Cordisburgo, em Minas Gerais. Próximo, por favor. E outros carbonatos retrabalhados também da Chapada Diamantina, que são belíssimos, né? Alguns com cambuqui, com laminações cruzadas por onda. Então, assim, existe uma diversidade de faces enorme dentro do mundo carbonático, justamente por causa dessa grande variedade, né, da forma como o mineral carbonático é precipitado e o que acontece com ele a partir do momento que ele se forma no ambiente marinho, por exemplo, que é o mais comum. Próximo, por favor. Mas não só de complexidade de faces se vive o carbonato, mas também existe uma complexidade de genética enorme, né? Muito porque esses mesmos minerais carbonáticos são muito instáveis, são muito reativos e eles podem ser dissolvidos parcialmente ou totalmente. O mineral carbonático pode ser substituído por outro, né? Pode ter uma recristalização também total ou parcial. E aí, uma rocha carbonática que se formou como X, durante a diagênese, ela pode ser transformada totalmente para uma rocha Y, por exemplo. E essa transformação diagenética pode mudar muita coisa do ponto de vista físico e químico da rocha. Por favor. Bom, então entrando agora mais no que a gente vai falar, né, dos elementos trágicos e rochas carbonáticas, vocês vão ver que o que eu mais vou falar aqui é sobre esse grupo aí, dos elementos terras raras, por causa da enorme aplicação que eles têm em diferentes rochas, além das rochas carbonáticas. Então aqui vocês estão vendo o padrão de terras raras que a gente encontra hoje nos nossos oceanos modernos, na água do mar, dissolvido nos oceanos. Tá? Então a gente tem algumas características. Passa aí, por favor, Fabrício. Enriquecimento em terras raras pesadas sobre terras raras leves. Esse é o grupo dos terras raras pesadas, aqui, como a legenda diz, e aqui é o grupo dos leves. Então eu tenho uma inclinação positiva. Próximo, por favor. Os elementos lantânio, gadolínio e ítrio são enriquecidos com relação aos seus vizinhos, né? O lantânio aqui tem um pico de lantânio, uma anomalia negativa, por exemplo, de sério, e aqui uma positiva de gadolínio e positiva de ítrio. Então a gente vê que o padrão marinho ele é cheio de anomalias, né? Além da inclinação positiva, ainda tem a presença das anomalias. Passa mais um, por favor. E o sério, então, que eu me adiantei um pouco, né? O sério, nos oceanos modernos, ele apresenta uma forte anomalia negativa, muito porque esse elemento ele tem uma característica muito útil, muito específica, que ele se comporta diferente devido às reações redox do oceano, né? Se é um oceano oxidante, se é um oceano redutor, ele vai se comportar de forma diferente e aí vai formar anomalias positivas, negativas ou até mesmo ausência de anomalia. E como que esse padrão marinho se constrói? Essa construção dele, né, aquele padrão que eu estava mostrando ali, ele vai acontecer por diversos fatores. Esse é um tema longo, né? Mas é principalmente nos ambientes costeiros, quer dizer, na transição rio, estuário, delta, oceano. Ali, né, com o aumento progressivo da salinidade das águas e com as reações químicas que acontecem ali, o padrão de terras raras da água doce de um rio, por exemplo, que geralmente ele é mais flat, ele é mais horizontal, plano, ele vai sendo pouco a pouco transformado para aquilo que eu mostrei para vocês no slide anterior. Esse é um trabalho bem legal de 2006 que faz justamente isso, né? Dentro de um estuário, foi amostrando diferentes águas com diferentes salinidades e vendo como o perfil marinho foi sendo construído, né? Então, a gente vê uma mudança aqui, né? nessas bolinhas pretas aqui em cima. A primeira amostra é o padrão água doce, né? Zero salinidade. E a última aqui, ó, que é essa bolinha branca aqui embaixo, ó, que é uma forte anomalia negativa de sério, né, já é o padrão água do mar, né, com mais ou menos 35 ppm, né, de sais dissolvidos. Então a gente vê aqui, ó, uma mudança progressiva no padrão de distribuição de terras raras, com o aumento da salinidade. Né? A gente vai vendo como que as amostras vão se transformando até chegar no que a gente conhece como o oceano moderno. Né? As primeiras anomalias a se formarem são justamente as de lantânio e ítrio, né? E a anomalia de sério ela só se forma em salinidades mais elevadas, já mais próximo do que das salinidades do oceano. A gente vê que mesmo nessa aqui, ó, nessa amostra branca aqui com 8,45 ppm, você ainda não tem anomalia de sério, ela só começa a se formar nessas 3 amostras aqui com salinidades já bem significativas. Né? Então toda essa construção, ela acontece na transição, né, água doce, água salgada, tá? E esse mesmo padrão aqui, ou vamos dizer assim, bastante similar ao oceano moderno, né, a gente encontra em carbonatos desde o Arqueano, quase desde que a Terra é Terra, claro, com algumas exceções, né, porque o oceano Arqueano não era igual ao oceano de hoje. Mas, no geral, é igual. Então, quer dizer, essas reações no ambiente costeiro de transformação dos terras raras, né, de água doce para água salgada, provavelmente existem desde o Arqueano. Próximo, por favor. Então, rapidamente sobre o sério, né, por que ele é tão importante? O sério ele tem 2 estados, né, ele tem 2 estados de valência, 2 estados de oxidação. Ele pode ser mais 3 ou mais 4. Todos os terras raras são mais 3. Né? O sério é uma exceção, mais 3 ou mais 4. Né? E justamente por isso ele reage mais, né, às reações redondas. Então, o sério 3, uma vez em ambiente oxidante, ele vai ser oxidado para sério 4. Né? E essa reação de oxidação ela é catalisada pela presença dos óxidos de manganês, e são esses mesmos óxidos de manganês que vão capturar o sério 4. Se o óxido de manganês, que é uma partícula, né, é sólida, captura sério e leva sério embora da água, logo essa mesma água vai ficar empobrecida de sério, já que existe um ladrão, digamos assim, um ladrão de sério. Né? Então, por isso que o oceano moderno, atual, oxidante, ele tem anomalia negativa de sério, por causa da presença dos óxidos de manganês que estão levando esse sério embora. Né? Agora, se esse mesmo óxido de manganês, se ele sai da zona óxica e entra na zona redutora, especialmente a zona de redução do manganês, né, esse sério vai ser reduzido, né, a partícula, o óxido de sério, ele vai ser reduzido, vai passar por uma dissolução redutiva e vai liberar esse sério capturado de volta para a água. E aí a gente pode ter dois reservatórios. Um reservatório superficial oxidante com anomalia negativa de sério, como essa figura tenta mostrar, né? Uma anomalia negativa, então menor do que 1. E outro reservatório redutor com anomalia positiva de sério, por causa dessa liberação de sério com a dissolução dos óxidos de manganês. Então toda essa brincadeira que eu estou falando, né, é justamente a aplicação, né, das anomalias de sério para se identificar oceanos redutores ou oceanos oxidantes. Vocês vão ver aqui nas próximas palestras, né, que muito desse assunto de oxidação dos oceanos, disponibilidade de nutrientes, surgimento da vida complexa, tudo gira em torno do oxigênio. Oxidação, né? Oxigenação da atmosfera e oxigenação dos oceanos. Tem muitas ferramentas para se distinguir isso. E as anomalias de sério é uma delas. Tá? Próximo, por favor. Então, eu posso ter anomalias negativas e positivas de sério, se, claro, né, houver oxigênio dissolvido nos oceanos, suficiente para formar óxidos de manganês. Bom, então eu vejo aqui o padrão água do mar, que eu já mostrei para vocês, né? Inclinação positiva, anomalia positiva de lantânio, negativa de sério, positiva de ítrio, padrãozão água do mar. Se eu olhar em diferentes materiais, em diferentes reservatórios, eu não vou ver padrão similar. Né? Tem coisas completamente diferentes da água do mar, aqui, ou aqui em cima. Mas tem um desses materiais que é bem parecido com a água do mar, que é calcita, carbonato altigênico, digamos assim, pode ser uma dolomita, né? Quer dizer, carbonatos formados in situ, na água do mar. Esses carbonatos, então, o sinal de terras raras deles acompanha, preserva o sinal de terras raras do oceano do qual ele se formou. Tá? O sinal de terras raras também, então, ele pode ser encontrado em rocha total, né? Quer dizer, eu pego a rocha carbonática, dissolvo ela completamente e a partir desse material eu consigo resgatar esse sinal marinho, pode ser em rocha total, ou pode ser, por exemplo, só na micrita, só na lama carbonática, pode ser em microbialitos, pode ser em grãos, como, por exemplo, um moóide, um intraclássico, um teloide, né? Mas em conchas e carapaças, que eu falei lá atrás para você, né? Por exemplo, a concha de foraminífero, ostracoide e tantos outros, né? Gastrópode, esse material pode sofrer com fracionamento orgânico. Lembra que eu falei que esses organismos eles têm a precipitação de carbonato de cálcio dentro das células, né? Para justamente formar essas carapaças e essas conchas. E esse tipo de processo, muitas das vezes, fraciona isótopos e elementos. Né? Então, alguns organismos, né, algumas conchas, algumas carapaças são úteis, né, no caso dos terras raras, outras nem tanto. Né? É um pouco dependente de espécies. Tá? Bom, e aí como é que, né, um geralzão assim, como que eu vou trabalhar isso? Né? Eu vou, primeiro, obter o dado, seja por dissolução parcial, dissolução total ou laser ablation. Depois eu vou corrigir esses dados, que é algo normal para qualquer análise química ou geoquímica. Depois eu vou normalizar para um material de referência. Vocês vão ver aí nos próximos slides que todos os gráficos que eu mostro no eixo Y é sempre normalizado. Né? É assim que a gente trabalha com terras raras e com tantos outros elementos e isótopos. E aí, então, finalmente, eu tenho um padrão de distribuição, eu tenho as anomalias e posso começar a interpretar aquele ambiente sedimentar, né, aquele oceano ou aquela bacia na qual o carbonato se formou. Passa, por favor. E aí, então, eu vou ter ali um padrão de distribuição, né, aqui no caso estromatóide devoniano, e aí eu vou ter diversas informações para interpretar. A concentração total de terras raras, razão intriônio, anomalia de lantânio, de sério, de európio, de gadolinho, razão para iodina interno, enfim. Não vou explicar cada uma delas aqui, mas são várias as informações que eu tiro a partir desse caminho que eu faço. E é a partir desses dados, dessas informações, que eu vou interpretar sobre, a condição química e bioquímica de formação dessas rochas. Próximo, por favor. Então, aqui, outros dois exemplos, microbialitos do Cambriano, por exemplo, ou microbialitos do Arqueano. Né? São duas rochas orgânicas, muito interessantes, né? Mas com um padrão de distribuição de terras raras completamente diferente. Pode ser uma questão simplesmente do estado de oxidação daquele oceano, ou pode ser uma questão de genética, de preservação do sinal, né? E tem também, claro, uma questão que são tempos muito diferentes. Uma coisa é o oceano há 3 bilhões de anos atrás, outra coisa é o oceano há 530, 520 milhões de anos atrás. Né? Então, as diferenças podem ser muitas por diferentes fatores. Próximo, por favor. Outro exemplo aqui, estromatólitos gigantes do Permiano. Né? A gente vê uma confusão de sinal, uma confusão de informação. O que é essa confusão? Né? O que essa ausência de padrão, digamos assim, significa? Cada estromatólito, cada amostra foi formada em uma bacia diferente, ou melhor dizendo, em uma condição bioquímica diferente? Pode ser que sim. Ou será que a diagênese atuou de forma um pouquinho diferente em cada uma dessas amostras? E por isso eu tenho essa confusão. Ou será que é uma questão do método? O método para se obter a concentração de elementos terras raras não foi o mais adequado? Pode ser também. Né? Então, todas essas variáveis precisam ser percebidas, precisam ser analisadas para que a gente tenha uma resposta. Se essa variação é uma questão ambiental ou não. Então, preservação, né? Eu introduzi um pouco esse assunto já, mas a preservação do sinal primário nos minerais carbonáticos, né? Qual é a robustez dos elementos terras raras e outros traços durante a diagênese, né? Como eu falei lá atrás, a diagênese em rochas carbonáticas ela pode ser muito intensa e pode apagar o sinal primário completamente. Né? O que acontece com esses elementos quando, por exemplo, aragonita e calcita de alto magnésio são neomorfizadas para calcita de baixo magnésio, que é um caminho perfeitamente normal, perfeitamente natural. E se não parar aqui na calcita de baixo magnésio e avançar para a dolomitização? O que acontece? Será que a dolomitização apaga o sinal total, parcial? Não acontece nada? Essas são perguntas muito boas, mas de respostas muito difíceis ainda. Não existe uma resposta absoluta para isso ainda. Existem vários trabalhos, várias discussões, e a gente vê que esse caminho pode apagar o sinal completamente, mas não também. Depende. Depende do contexto diagenético, depende do contexto deposicional, depende da química do fluido diagenético, da quantidade de fluido diagenético. Então, não, aqui não existe nem sim, nem não. Existe um depende. Então, aqui a gente vê uma figura super interessante, é um trabalho fantástico que existe, né, de 2019, que mostra a evolução do padrão de elementos terras raras à medida que a gente avança para as zonas redox no soterramento desse sedimento, né? Ou seja, durante a litificação desse sedimento. Então a gente vê como que, aqui em vermelho, o padrão de terras raras, como que ele pode ir mudando à medida que o sedimento vai afundando durante o soterramento. Ou às vezes mesmo dentro de uma esteira microbial de poucos centímetros, né? Que é um sistema fantástico. Então, a diagênese, né? O que essa figura mostra? Vamos resumir em um minuto, né? Que é o tempo que a gente tem. E a diagênese pode claramente modificar o padrão de terras raras de carbonatos C. E aí tem um C. Né? Se o fluido diagenético tiver a assinatura de terras raras muito diferente do fluido deposicional, então a gente já imagina mais ou menos que uma diagênese precoce, uma eodiagênese, vai afetar pouco, justamente porque o fluido ainda compartilha muita característica com o próprio fluido deposicional, e que talvez uma mesodiagênese, uma diagênese tardia, é que ela sim pode, né, alterar, modificar e, vamos dizer, danificar o sinal primário. E claro, se a razão fluido-rocha for alta, quer dizer, se houver bastante fluido nesse sistema diagenético. Tá? Então, a quantidade de fluido e característica química do fluido. Se essas duas coisas forem alcançadas, aí sim a rocha pode, né, a rocha que você coleta no afloramento e analisa essa rocha, pode não ter, o sinal químico isotópico dela pode não ter mais nada a ver com o ambiente deposicional, mas sim com a diagênese. O que para alguns, para muitos, na verdade, é bastante interessante. Estudar a diagênese tem muitas funções, né, inclusive funções econômicas, né? Por exemplo, reservatório de petróleo. Próximo quadro. A mesma coisa dos terras raras vale para os outros traços, tá? Qualquer um dos outros traços. As mesmas premissas, característica química do fluido e quantidade de fluido. Então a gente vê, né, diferentes materiais que capturam elementos na água do mar. Por exemplo, materiais orgânicos que capturam cádmio, cobre, níquel, zinco, que são micronutrientes essenciais, que certamente serão abordados aqui. Óxidos de ferro que capturam na água vanádio, antimônio, arsênio, né? E manganês que captura uma infinidade de elementos, e um dos maiores ali é o molibdênio, né? Que tem grande importância também para essas questões redox e que deve ser abordado aqui ainda hoje, né? Então, esses materiais, uma vez que são soterrados, né, no sedimento e vão para profundidades maiores, eles também podem ser liberados pelo fluido diagenético e, com a precipitação de novos minerais diagenéticos, esses minerais podem ser enriquecidos em cromo, cobre, zinco, molibdênio, arsênio, né? E essa zona aqui, ó, que eu até nem falei no slide anterior, mas essa zona de redução do ferro e do manganês é uma das zonas, né, que mais fornece elementos para o fluido diagenético, né? Lá atrás, volta o slide, por favor, rapidamente. Né? Essas esse enriquecimento nos terras raras médio, que dá essa corcova, né, essa corcova do padrão de distribuição, é bem característico da zona de redução de ferro e de manganês. Né? Próximo, por favor. Então, mais uma vez, né, a dissolução, oxidação, remineralização desses materiais aqui pode sim modificar completamente a assinatura química de uma rocha carbonática durante a diagênese, se aquelas premissas, como eu já falei, forem atendidas. Então, indo já para a parte final, né, brevemente, outra questão que eu acho que vale a pena ser abordada e que é de extrema importância é o método com o qual a gente obtém esse dado químico. Né? Não é qualquer método que funciona, não é qualquer método é sempre adequado para qualquer situação. Existem, hoje em dia, basicamente três, né? Que é a dissolução total, dissolução com multiácidos, clorídrico, nítrico e fluorídrico, ou a dissolução parcial, usando ácido acético ou nítrico, ou laser ablation. Né? Não existe um método errado e um método certo. Cada método vai te dar uma possibilidade diferente de interpretação dos resultados e de investigação. Próximo, por favor. Rapidamente, dissolução com multiácidos. Então, esse é um método que eu posso ter problema com contaminação detrítica, porque eu vou dissolver a amostra completamente. 100% dela será dissolvida. Se tiver ali 1, 2, 3% de argila, de feldspato ou qualquer outro mineral que não seja carbonático, óxidos, sulfetos, e a química desses minerais será também calculada junto com os carbonatos. E esses minerais, muitas vezes, eles não me interessam. Pelo menos aqui, tratando de traços de terras raras, eles não me interessam. Então, 1, 2, 3% desses minerais na rocha já pode ser suficiente para mascarar o sinal primário químico da bacia ou do oceano. Então, é necessário triar os carbonatos mais puros, né? A partir de diagramas binários, eu vou triar os carbonatos mais puros e usar somente esses para interpretações paleoambientais. Nesse diagrama, por exemplo, eu usaria somente essas amostras aqui, ó, que estão próximo da origem, quer dizer, um somatório total de terras raras bem pequenininho, somente esses aqui são os carbonatos mais puros. Carbonato muito puro, né, ele sempre vai ter concentração baixa, geralmente, quase sempre, ele vai ter concentração bem baixa de terras raras. Tá? Então, ali é o meu interesse para fazer interpretações paleoambientais. Aqueles com maior impureza, bom, eles vão ficar para outras questões, por exemplo, proveniência, estratigrafia, quer dizer, quando que teve mais aporte detrítico, quando que teve menos, então, enfim, eu ainda posso usar, mas para outras questões. Próximo, por favor. Só para ilustrar, né, aqui a gente tem uma amostra totalmente pura de carbonato, né, essas aqui, essa mais de baixo aqui em branco. À medida que você vai aumentando a concentração de detríticos, por exemplo, né, se eu comparar a amostra 100% pura de carbonato com a de 2% contaminada, veja, a anomalia de sério já foi embora, eu não vejo mais, e a anomalia de nítrio-ômio, ela também quase não existe mais. Então, com 98% carbonato e 2% detrítico, já é suficiente para eu não enxergar mais o sinal marinho, não enxergar mais o sinal primário. Por isso que 1, 2% já pode realmente bagunçar a coisa. Próximo, por favor. Então, é necessário trabalhar com carbonatos de altíssima pureza, né? Ou utilizar outros procedimentos analíticos. Nem sempre na bacia que eu estou trabalhando os carbonatos são puros, às vezes não dá essa sorte. Então, como contornar isso? Muda o método. Uma opção é a dissolução parcial, com ácido acético ou nítrico. Esse já é um método que é amplamente utilizado hoje em dia, né? Eu uso ácidos fracos, diluídos, que vão dissolver quase que só carbonato de cálcio ou só carbonato de cálcio. Né? Então eu consigo dissolver, somente carbonatos, ele é útil para fugir dos carbonatos contaminados, quer dizer, mesmo que tenha ali 2, 3% de argila, o ácido é tão fraco que vai dissolver somente carbonato e não a argila. A argila vai permanecer lá onde ela está. Então, o que eu vou levar para o espectrômetro para ser analisado é somente carbonato de cálcio dissolvido. Eu posso, dependendo do tempo de reação e da concentração do ácido, eu posso dissolver somente algumas frações, 10, 20, 40% de carbonato, mas eu ainda, por exemplo, tenho aquele problema da diagênese, que eu não sei qual carbonato está sendo dissolvido, desculpa. Eu não sei se é o cimento, se é o grão, se é a micrita, se é a carapaça, eu só sei que é carbonato de cálcio. Mas tem muitos carbonatos de cálcio diferentes. Né? Rico em ferro, pobre em ferro, alto em magnésio, baixo em magnésio, aragonita, não sei. Então a questão da diagênese permanece complexa nesse método, mas resolve o problema da contaminação. Aí entra o último, que é o laser ablation, agora é uma análise in situ, né? A partir de um microscópio, a partir de um laser, né? Um peixe de laser de poucos micrômetros, 50, 100 micrômetros, eu consigo, então, amostrar áreas micrométricas ou milimétricas específicas. É claro, eu preciso ter um bom controle micropetrográfico, então eu preciso sentar no microscópio durante horas e horas até identificar as zonas que eu quero amostrar, os locais que eu quero amostrar. E com esse método eu posso identificar assinaturas geoquímicas específicas, por exemplo, cimentos diferentes, se é o cimento mais primário ou mais tardio. Eu posso identificar a composição química da micrita, de grãos, por exemplo. Não, eu só quero a composição química de oóides. Beleza, o laser ablation pode te dar isso. Eu só quero a composição química de carapaças de ostracode. O laser pode fazer isso. Ou alguma espécie específica, um grão específico. Mas, claro, ele amostra tudo que tiver dentro da área do spot, dentro do feixe do laser. Se esse feixe for grande, de 100 micrômetros, e o que eu quero amostrar tem somente 10 micrômetros, não vai funcionar. Né? O spot é maior do que a minha área que eu tenho interesse. Então, essas coisas têm que casar. Eu não consigo também fazer tudo. Né? Mas é uma possibilidade interessante para fugir da contaminação detrítica e também para amostrar, né, fases diagenéticas ou grãos específicos. Por favor. Então, aqui mais uns exemplos do laser ablation. Né? Eu tenho esse artigo de 2015, ele é bem interessante também, que ele mostra aqui uma franja de aragonita bem primária, né, e uma calcita blocosa no meio. Então, as franjas bem primárias, com sinal marinho bem preservado, né, forte anomalia negativa de sério, forte anomalia positiva de ítrio, inclinação positiva, quer dizer, idêntico ao oceano moderno, oxidante, bem misturado. Né? Enquanto que a calcita blocosa aqui, ó, observem, né, ela perde a anomalia negativa de sério. Isso aqui é ausência de anomalia negativa de sério, só que presença de anomalia positiva de lantânio. Por isso, né, parece que é anomalia negativa de sério, mas não é não. É positiva de lantânio no caso. Então, perde a anomalia negativa de sério e vejam que ganha uma corcova, né, um enriquecimento dos terras raras médios. Ou seja, muito provavelmente essa calcita blocosa ela foi precipitada na zona de redução do ferro e do manganês. E eu só conseguiria identificar isso se eu usar o laser. Imagina se eu tivesse usado, por exemplo, o ácido acético, que ia dissolver todos os carbonatos. O sinal de terras raras dessa rocha aqui ia ser uma média entre isso e isso. E eu ia mascarar o sinal primário, que está preservado somente nas franjas. Aqui é uma tentativa que a gente está começando agora, né? Então eu nem tenho muitos dados aí para mostrar, mas é dos carbonatos da formação Lagoa do Jacaré, né? Bastante oolítico e alguns microbialitos. Então esse aqui é só um exemplo para mostrar a seleção das áreas que vão ser amostradas com o laser. Próximo, por favor. Mais um exemplo do laser, né? Oóides do meio do ediacarano, né? Bem do tema que vai ser comentado aqui a tarde toda. Então, selecionou algumas áreas e conseguiu, né, principalmente aqui, dos oóides e das franjas bem primárias, bem eodiagenéticas que estão presentes ali, e o padrão de distribuição de terras raras, mostrando esse oceano ainda com pouco oxigênio dissolvido. E para encerrar, então, porque meu tempo já se esgotou, 2 minutos e eu termino, só um estudo de caso bem rápido, bem sintético, de microbialitos do Cambriano, ali do grupo Bambuí, norte de Minas Gerais, na Serra da Jaíba, tá? Que é essa serra que está na foto aí. Passa, por favor, Rodrigo. Então a gente tem um membro Jaíba, que ele pertence à formação Serra da Saudade, são microbialitos, são limestone belíssimos, tá? Trombolitos também muito bem preservados. Passa, por favor. Que estão aqui dispostos, né, mais ou menos 40 metros do membro Jaíba, um intercalado muito forte entre microbialito e alguns brainstorms, ambiente bem raso, provavelmente uma laguna protegida, né, e uma barreira oolítica. Então a gente fez diversas análises de elementos terras raras e outros elementos traços, e a gente chegou em algumas conclusões interessantes. Né? Existe uma contaminação detrítica, mas muito pequena, e que mesmo a dissolução parcial ainda pegou um pouquinho essa contaminação detrítica, foi usado um ácido clorídrico um pouco mais concentrado do que devia, mas apesar da pequena contaminação detrítica, o que mais chamou a atenção é que existia uma correlação muito forte entre os terras raras e o elemento ferro, né? Quer dizer, eram carbonatos ricos em ferro, e esse ferro, quase certamente, ele não é um ferro detrítico, mas um ferro altigênico. Porque o ferro, as concentrações de ferro, elas não correlacionavam a outros elementos que claramente têm origem detrítica. Então, existia um ciclo de ferro intenso na bacia durante a formação desses microorganismos. Passa, por favor. Então, qual foi a conclusão, no fim das contas? Né? Que são microbialitos depositados em ambiente costeiro, ainda numa zona de grande mistura de água doce e salgada. Né? Muitas das anomalias típicas do oceano não estão presentes nesses microbialitos, mas não por causa de contaminação detrítica e nem de diagênese, é a nossa conclusão. Então, talvez são um sítio deposicional ainda com uma água não tão salina assim, né? E aí a gente, né, criou aqui essa figurinha justamente para mostrar essas zonas de mistura e como que as anomalias e os valores de terras raras vão se desenvolvendo e onde mais ou menos o microbialito do membro Jaíba estaria posicionado dentro desse gradiente de mistura de salinidade. E agora a mensagem final, para encerrar, né, algumas conclusões. Né? Evitar carbonatos impuros, se possível, né, claro, e priorizar a dissolução parcial ou análises in situ, usando laser, por exemplo. Hidriônio, concentração de alumínio, zircônio, escândio, tório, todos esses são bastante úteis para informar a contaminação detrítica, e é sempre importante testar isso. Lembra que a contaminação detrítica é vilã nesse caso. Concentrações de ferro e manganês podem indicar precipitações altigênicas ou alterações pós-deposicionais. Né? É bastante importante ficar ligado nesses dois elementos. Avaliar não somente as terras raras, né? Não pode ficar preso ali, mas também outros traços e confrontar elementos. Né? E esse confronto de elementos, a gente só consegue chegar nesse ponto se a gente tiver uma visão abrangente da geoquímica. Né? Quer dizer, compreender os comportamentos dos elementos em condições ambientais e diagenéticas. E claro, para encerrar, é necessário dedicar um tempo compreendendo a fundo o contexto deposicional e petrográfico das rochas. Fazer estratigrafia de detalhe, lâmina drogada, e se possível, é sempre bom usar catodo, usar o MEV e tantas outras ferramentas úteis para a gente caracterizar melhor a petrografia dessas rochas e interpretar melhor, consequentemente. Muito obrigado, pessoal.
Okay, so we thank Gabriel, and if you got questions, we got like 30 people online, and there are some questions in the chat there, so that I think you can go into the chat and answer people directly, okay? I'm just going to do a quick reintroduction here for people that couldn't catch it before. So this is our second edition of the Project Mobile Day, where you have international visitors and our home professor here, Gabriel. And this is supported by Instituto Serra-Pilheira, CNPq, and our IMCT from CNPq, which is Instituto Geoatlântico. Então, é a nossa segunda edição do ciclo de palestras do Projeto Mobile, né? E a gente tem o suporte aí do Instituto Serra-Pilheira, do CNPq e do nosso Instituto Geoatlântico, que é também o Instituto Nacional de Ciência e Tecnologia do CNPq. A próxima palestra agora vai ser a professora Ying Zhu, pesquisadora da University College London. So, next stop now will be Ying Zhu, which is a researcher at University College London. Okay, Ying, and then when you want to start, let me just test this. It's supposed to be working now. Yeah, but is it working for like going back and forth, or it's just for the? Okay, so maybe we have to get it out here.
Não, o pendrive tá com ela.
Aqui a apresentação para o computador e ela vai começar um minutinho. So I think I'm going to be like jumping slides for you, okay? If that doesn't work, maybe that will work. Yeah, let's see. Got está escrito Minas Gerais. A lot of dots. Yeah. Okay. Vou até tirar e colocar de novo. Okay, you want to test that, the slides? It's not going back and forth, right? I think it's only the laser pointer is working. Esse é o business vai funcionar. Maybe start and then try this one. Okay, I'll try this one to see if it works, but you can start on top.
Hello everyone, my name is Ying, and I'm having a cold, so I might stop my talk in the middle and cough crazily. Anyway, so I am a sedimentary geochemist and currently work on mostly Tonian new Proterozoic and some Mesoproterozoic rocks. I'm interested in practice on weathering and also on redox. Not only I'm interested in to produce data, but also quality data, which means I'm also interested in method development. So today, this talk, I'm going to present some data from South China, bring you to another craton during the late Ediacaran. Okay, so during the Shuram excursion. Oh yes, the title has slightly changed. I'm going to talk about the redox evolution too, other except for the role of nutrient feedbacks. So many of you might already have known that the Ediacaran period is a very eventful and period during Earth's history, and it just happened after the snowballs Quarantinian, after Marinoan. Yes, that's no. Oh yeah, oh sorry. Yes. Yeah. Okay. Sorry. Yes, and also have quite a few the delta carbon 13 negative excursion and a lot of biological innovations during this about 100 million years, okay, and of which, and the most mysterious and long-lasting and with great magnitude of the delta 13 carbon of carbonate. Its negative excursion is called the Shura-Vanuka excursion, with which the delta 13 carbon go down as low as -12 per mil, and correlate with which is the current bywater. Here, this excursion and the bywater have been found on numerous continents, Russia, Newfoundland, Canada, Namibia, Oman, and so on. And today I'm going to take you to South China. Okay, so the South China has well-developed the current successions. Okay, and one correlable negative delta 13 carbon negative excursion is called DONS, named by Professor Mao Yanzhu here. Okay, and Professor Zhu has done extensive studies of multiple sections of the DONS in South China, all have shown the negative excursion. And today, I'm going to focus on the DONS correlate with the Shura, okay, and the recovery part from the negative come gradually back to zero. This black shell part. Okay, here. So here we also found the Onhui and the Miaohe biota. Okay. So normally with a negative excursion, people start to think oxygenation, oceanic oxygenation event. Is the Shura excursion one oxygenation event? People have, with this question in their mind, people have started. Okay, here I'll show you a few examples. So 2013, Sahu et al. Used the redox sensitive element enrichment and has identified about 4 oceanic oxygenation events. Here we can see with the spikes or enrichment of the redox sensitive elements, including vanadium, uranium, and neptunium and uranium. Okay, and one of which this one is correlate or from the black shell I'm going to present you with. Okay, has been identified as oxygenation event. Moreover, Fifi Zhang has used uranium isotopes, okay, to study this negative excursion. The uranium isotopes shows basically the toxic ocean seafloor over the total seafloor. And this data shows you that during the Shura, the uranium isotope ratio is the same level as the molten Bahamian carbonate, indicating similar oxic seafloor ratio over the total seafloor ratio during the current period to today, meaning oxygenation. Okay, so there are quite a few hypotheses which will try to investigate what has caused the Shura excursion. Okay, and some people don't even believe it's real, since it's diagenesis effect. Okay, some talking about oxygenic carbonate model, some suggested missing. And here, by one of our next speaker, Professor Shields, has combined the sulfur cycle with carbon cycle, which answers not only that the duration of the Shura, but also the magnitude of the delta 13 carbon. Now, remember I said the largest -12 per mil to 0. So that is the largest one. Here with this model, it tells us that with extra evaporite weathering, bringing new sulfate into the ocean. Okay, the extra sulfate into the ocean, into an anoxic ocean, which started to precipitate as pyrite, and this process produced oxidant or oxygen, which will oxidize the dissolved organic carbon. Okay, this will introduce the 12 carbon rich carbon into the seawater, which will be precipitated as the carbonate. That is how the Schiller excursion has been produced. But another thing is that the oxidation of the DOC will produce extra CO2 released to the atmosphere, and this will enhance the chemical weathering, okay, which means extra, the whole feedback, positive feedback, bringing more evaporite and possibly nutrients, which we'll discuss later. Okay, if this model is right, okay, like we were doing the detective story, if this person is murdered, now we have a hypothesis. How can we prove it? Okay, so we have to show some evidence. So, if the deep pyrite burial really oxygenates the ocean during the Schramm excursion, and do we have evidence for this pyrite burial? And if euthymia, which is sulfidic condition, okay, was widespread, where is it? In the basin, okay. Of course, I'm not the most smart one because people already start to think about it. There are some studies already happened. So, to look at euthymia, I think the best proxy is iron speciation. I think Fabrizio is going to build the technique here, the lab. Maybe next year you'll already be able to produce your own iron speciation data. So this is a sequential leaching technique, which look at different iron pools. So how iron is fixed in a sediment. Okay, we look at the sulfide and now bound with iron, and the carbonate associated iron, and iron oxide, and the magnetite pools. And because it's an empirical technique based on modern sediment, so we have a couple. Of course, this needs to be discussed or possibly updated someday. So basically, the four pools form the highly reactive iron. The highly reactive iron over total iron is over 0.38, and this is indicative of an anoxic water column. But on the contrary, if this ratio is smaller than 0.22, then the water column is oxic. So if the highly reactive iron over total ions over 0.38 anoxic, and then we look at another ratio, which is pyrite iron over the highly reactive iron pool. If it is over 0.8, consider that the situation is a euthymia condition, okay? And whereas the pyrite over highly reactive iron is smaller than 0.8, we call it a fruitinous condition, iron-rich condition, okay? And between these ratios, we think it's equivocal, possibly euthymia. So there are other, of course, proxies can help you decipher this. Okay, so as I said, people already started to look into this in different sections. As early as 2010, Li Chao has looked at one of the prominent or the type section, Geelong 1, and draw the conclusion that the current ocean is a stratified ocean with euthymic wedge. Okay, but back then he thinks this Geelong 1 is on the whole basin. Later we'll update on that. Okay, then off FL 2016 look at the central number 4, and this time they did look at the different basins, the intershell and the slope and so on. And what they see is again a wedge in the intershell basin. Okay, and much more studies, but again on the Geelong 1 and the Yangtze Gorges core shows indicating anoxic condition on both and the euthymic condition. Li Chao updated his study in 2015, look at another two sections, Miao and Trina. Here you can see mostly euthymic with episodic ferruginous conditions and one possible suboxic condition where the Miaopu biota was found. And the 2019, the Ostrander updated the deep slope basin, again, also combined in the even isotope study. Here it also shows the sample number 4 is mostly eucinic, anoxic eucinic. Okay. And other few ones are deep. Interestingly, their target, what they look at is a deep slope section. It's mostly anoxic, but ferruginous condition. So what this shows you? Shows you that the eusenia is indifferent, okay, in the studies, in the lagoon and slope settings. But it's still not very clear where exactly they are. So that is how my project comes in. So we'll combine, of course, the published data. So we're looking at another 7 sections across the whole basin. Here you can see where they are. 3 lagoonal settings, 2 upper slope sections, and 2 middle slope sections. Here's what has been published, okay, all these sections where they are, and where the new sections, the new data are coming from, okay? So we hope we can cover the whole basin, so that we know to answer these questions. Okay, these are the data. Just quickly go through, just to lead. Yeah. To lead your eye, the yellow color is eucinic, the black color is, yellow color is ferruginous, and the black color is eucinic. So this one section called Shanghai 1, the lagoonal setting is mostly ferruginous, okay, and we have episodic eusenia at the top of the sample number 4, and these data correlate with higher TOC and a high carbon to phosphorus ratio, and this we will discuss in more detail in the second part of the talk. Okay, and also we can see the enrichment of some of these redox sensitive elements, uranium, vanadium, and thorium. Okay. And the next one is another lagoonal setting. Here shows you mostly ferruginous, as we can see here, and sporadic eusenia, and correlates also quite well with the high TOC, this black color, and see the carbon phosphorus ratio is a green color. You can see they're quite high, over 1,000. The next one, the clinical setting, another lagoonal setting, here is different. Okay, they have slightly depth difference. So from basically the dose number 4, the black shows from ferruginous to mostly eusinic condition with possibly one wave of oxic condition. Okay, and this part correlates quite well with the higher TOC and higher carbon phosphorus ratio and enrichment of RSE. So these 3 new lagoonal settings shows you that eusenia correlates quite well with productivity. Okay, so the upper slope section 110. Here we have not only the black shell, but also the overlying Liu Chapo formation, which is equivalent to the terminal, the Dunning formation, if you know of it. So what we can see is the doshan number 4 is mostly eusenia, the black color, and with one episode of ferruginous. But the Liu Chapo is mostly ferruginous condition, okay? And if you can see it it's not very clear. The doshan number 4 has much higher TOCs, up to 3%, but the Liu Chapo has 0.3%. Okay, so there's a huge difference of productivity, and the carbon phosphorus ratio is also massively different. Here is up to 400, here is up to 30. Okay, ratio. So the Shajiao section, I have one more study later on this section. It covers the whole of the central formation. Here, what it shows us is the lower part of the central formation is mostly fusionous, and from the Schumann excursion, for example, level 3, we can see mostly Eusenia with episodic fusionous condition. And if we come to the central number 4, the black shell, you can see it's mostly Eusenic condition with one with oxic condition. And these correlate very well with high carbon phosphorus and TOC and the in vitro of the redox sensitive element. Again, Taoyin middle slope here is very interesting. So basically, we have mostly started the fusions conditions, mostly Eusenia with a few possible oxic conditions. Then the upper dosage number go back to mostly fusions conditions, and the Eusenia correlate, you can see very well with high TOC, say up to 12%, and the CP ratio up to 4,000. This is massively high. And here is also where the quite famous fossil, the Euandromeda octabracciata. So again, Professor Drew's finding, and the group has been reported and been found. Okay, the Fontaine section is mostly Eusenic, as you can see here. Okay, and the upper section is mostly silicate class A interbedded with shells, it shows mostly fusions condition with one possible oxic condition. And the Dexter shell again correlate with high TOC and CP ratio. So what does that tell you? What does that mean? So let's go back to the whole base law reconstruction. Let's superimpose. This is published data, okay, and what we have found, okay, and this I will translate for you in a very ugly drawn schematic, okay. This is the conclusion. So basically, so basically you have episodic Eusenia in the slope lagunal settings and continuous Eusenia in the deeper lagoon settings, and the continuous Eusenia in the middle slope settings of the open Nanhua basin, and possibly a Eusenic wedge. And this has been confirmed with our study of molybdenum isotopes. Molybdenum isotopes are possibly, Gabriel just also showed some. Basically shows you not only the redox condition, but also if the basin is restricted or connected. Okay. So here our data, molybdenum isotope data here confirms the multiple oxygenation events during the whole ocean formation, but also our data compiled with published molybdenum data shows you that enrichment of molybdenum in the lower dosage number 4 and the highest molybdenum isotope ratio on the top of the dosage number 4. This gives us two indications. One is the oxygenation, of course. This is the peak molybdenum isotope ratio, and the published uranium isotope ratio from Kendo indicate that this is very close to Morton level, indicates oxygenation. But at the same time, these possibly indicating the maximum flood level at the beginning of dosage number 4, and the starting of the regression only at the top of dosage number 4. These two, we can discuss that if you are interested in this concept later. Okay, now we answer this. We have three questions started. Have we answered them? Oh. So we did find pyroburyl sulfate, reduce the sulfite, and will give 2 molar of oxygen. So we could answer the first question already. Second, what is the evidence? And we have seen loads of the paraburial evidence that widely spread leukemia. We also showed you where the leukemia was with my schematic. So remember I said that the another limb of this cycle, when the oxidation of the DLC will produce extra CO2 to the climate, into the atmosphere. It's like today, we have a warm climate. Okay, we have intense chemical weathering will bring in extra nutrients and extra evaporate into the ocean. And then in this case, what role did the nutrient cycle play during the sugar? Okay, will this form a positive cycle, positive feedback, or negative? Okay, with this we started to look at another sequential leaching technique of phosphorus, okay, from the same lab. So what it shows you, so as we know how phosphorus ends up in sediment, okay, through the riverine input and so on, from detrital, apatite, organic matter, and iron-bound phosphorus, okay, coming to the ocean. In Arctic Ocean today, so 90% of the organic carbon will be remineralized and release back the phosphorus into the water column. Okay, and here we're looking at the different pools. Okay, yes. So, but under fruitiness conditions, remember what I said? Fruitiness
A pilha acabou.
So under fruitiness conditions, which is iron rich, okay. So the same become a much more sink to phosphorus in the fruitiness ocean. And the bacterial accumulated and organic matter. But under the acidic condition, the phosphorus is recycled back to the water column during the iron mineral reduction by hydrogen sulfide. Okay, remember we said the pyrethrum. And also the preferential release of phosphorus from decaying of organic matter through sulfate reduction. Okay, so it's a whole process. Remember sulfate reduction, pyrethrum, uracemic. That is how you relate it to the phosphorus cycling in the ocean, which is a limited, critical nutrient in the ocean. Okay, and here we can see, therefore, we have found out, and of course, during all of these redox conditions, another phosphorus sink we cannot ignore, which is this phosphorus lock-in or formed during the oxygenic mineral formation. So mostly is, I think it's fluoroapatite formation and also iron phosphate. Okay, and this process. So this whole sequential process is basically to single out the different pool of phosphorus. Okay, so we have now the total phosphorus formed of these four pools: iron, the ferric hydride, and oxygenic mineral related associated phosphorus, and organic bound, and this basically is how when the phosphorus come in as a detrital appetite, for example, into the ocean. Okay, so how can we separate that? Through this very convoluted and complicated process. And then besides, under the euthymic condition, remember what I say, the phosphorus is readily recycled back to the seawater. And as a consequence, what happens is the CP ratio, remember we keep talking about in the last part of my talk, is the CP ratio is normally multiple magnitudes higher than the modern reactive ratio, which indicates high productivity. Okay, so yeah, of course, I got this. So the reactive phosphorus is this 3, except the tritally associated phosphorus. Okay, I'm not going to show you all of the sections, but as only 2. Here we can see the phosphorus speciation and shows you the correlable redox condition of euthymic. You can see the enrichment of organic bound phosphorus and the other part, but the detrital is this gray color, and one oxygenic phosphorus and also iron bound. You can see the CP ratio both are quite high in places and as high as 2000, much higher than the Redfield ratio, which is 106 over 1 or 150. Okay, the next one, as an example of the deep red slope section, here shows you also the different phosphorus associated with different phases. Okay, and what that shows you is the high phosphorus concentrations are probably due to elevated organic content. Okay, so the TOC, remember, and the significantly higher sediment phosphorus aluminium ratio compared to the average shell value, and we have very high organic carbon over reactive phosphorus and organic carbon over organic phosphorus model ratios in euthymic conditions. Okay, this bit, the lower part. If we look at these 2 ratios, it's basically this shows you the extent of phosphorus either fixed in a sediment or released back, being recycled to promote the primary productivity. Okay, and here we can see the modern Redfield ratio is this red line, and we can see most of our samples here, including some Fujian's conditions, they are mostly over the Redfield ratio, meaning high productivity. Okay. So, what role did the nutrient play during the Schuart? Okay, here we can see a very simplified schematic, and you only look at the phosphorus part of the cycle, ignore the nitrogen. So basically, the primary productivity increases. Okay, this is a positive feedback, and it increases anoxia on the seafloor. Okay, and then this will reduce the iron oxide-related phosphorus burial. Remember, I said during the euthynia, phosphorus is readily released from the iron-bound minerals back to the water column, and with the help of reduction by what is called hydrogen sulfide. Okay, and this again will reduce the burial or the fixation of phosphorus in the sediment, okay, which means more phosphorus will be in the seawater column and promote more organic matter production. Okay, so this basically means, but it can go other way too. Okay, if you look at the cycle other way around. So I think we have 4 questions to start with. And we'll answer the first 3 with the first part of the talk. And the 4th question, I hope I have answered you. As basically, the first part is showing iron speciation shows widespread leukemia that would have promoted pyrite burial during the Shuang anomaly early zones. Anomaly in South China, and the phosphorus data shows how extensive phosphorus recycled during the latter part of the Shuang may have sustained the productivity, seafloor anoxia, and the pyrite burial. That's it. Thank you very much.
Thank you, Wayne, for the very interesting talk. And we do have time for questions. If you guys want, someone want to ask a question. Yeah, I have a question myself. Like some of these settings that you show, they are like lagunal settings, right?
Yes.
And is there some kind of environmental constriction on interpreting like iron speciation data compared to like deep water sections and then lagunal sections? There's something that you have to take care of if you are doing these comparisons.
I don't think so. It is, even though we call it lagunal, it's a huge basin actually. I think it is just a part of the sea which is separated from the main basin. Yeah, I mean connected but different depths. Yes. Sorry for my broken parts of contact. Yeah.
Okay, so if there's no more questions, we'll have a coffee break now, okay? And then we'll be back at, I think, 3:45 maybe. So yeah, now we're on time. Yeah, around 3:45 we're back with Eric and Graham. So coffee break agora lá fora e uns 15 minutos a gente volta, tá bom? Obrigado, pessoal. So are we live now? Okay, so we're back, and now we're gonna have Eric Sperling from Stanford University, who give us a talk about his work, the project there. Então a gente voltou e agora o Eric Sperling, que é professor lá em Stanford, trabalhando em colaboração com a gente aqui, vai dar uma palestra sobre o projeto deles que estão andando.
Great. Thank you so much. This is my first time in Brazil, and it's been such an awesome trip. And I'd really like to thank Fabrizio and Gabrielle and Lucas Warren for leading such an amazing field trip to the Manly Group. So today I'm going to be talking about kind of two different things that roll into each other. The first is kind of more generally how we treat data in the geosciences or the paleogeosciences, and then I'm going to follow on from Dr. Hughes' talk about questions of oxygenation around the Dyack and Cambrian boundary. This is my field area in Northwest Canada. I feel really lucky to work there. It's such a beautiful place, such a great rock record at the Neoproterozoic and Paleozoic. And a couple of years ago, I was able to work at this absolutely outstanding section of the Road River Group on the Peel River. I had a video. Unfortunately, I had some PowerPoint issues, so I had to turn my slide into a PDF, so I can't show you this video. But it is one of the most spectacular sections, it is the most spectacular section I've ever worked on in my life. You can start in the Upper Cambrian and walk all the way to the Middle Devonian with basically no faults, no breaks in section, and no changes in depositional faces. I've never seen anything like it in my life. So I did this work with Justin Strauss, one of the best sedimentology stratigraphy people of his generation. We measured the entire section, got all the grapholite fossils, took a bunch of geochemical samples. We brought it back to lab. We did a bunch of these ion speciation analyses that we were just introduced to. And we created a plot like this. Don't worry about the data here. This is basically just our stratigraphic column going through the Ordovician, Silurian, and Devonian. These are ion speciation metrics. The important thing here is we see points moving from the green field, which indicates that bottom water conditions were anoxic but not sulfidic, or what we call ferruginous, up more into this orange field, which is the presence of sulfide or euxinium. We see similar things when we look at molybdenum and molybdenum-uranium ratios. So we were all really happy with this story. It has a lot of interesting things to say about how early land plants affected the Earth system. But that's not really the point of today's story. What I want to introduce here today is that this is by far the most spectacular section I'll ever work on in my entire geological career. But this only covers about 2.6% of Earth history. Well, similarly, if you look at the Bamboo Group, the rocks that we looked at on the field trip, a great record of the Ediacaran, but still just kind of a couple of snapshots of the Ediacaran. The other thing is that those Bamboo Group rocks are recording a very different depositional environment than what we're looking at in the Road River Group. Even within one depositional area, you have big differences in the rocks simply based on where you sample. So these show the same rock unit, the Permian Wolfcamp Formation from West Texas. Here you see a core. It's organic, it's really dark, right? Has a ton of organic carbon and pyrite in it. This is what it looks like in outcrop. It's absolutely oxidatively weathered. It's lost all of its pyrite and organic carbon. And there are geochemical samples from both of these that are published in the literature. So if we want to be comparing data through geological time, the clear thing that we need is that we need a big data product of all of the geochemical data through geological time, but we also needed to have all of this relevant geological metadata, so the depositional environment, whether it's from a core outcrop, the latitude and longitude, all of this geological metadata associated with it. And quite simply, we don't have that. And so if we want to be looking again at questions of paleoenvironmental evolution over really long timescales, what we were left with was we needed to build this data set. And so this led us to think about some of the different ways that different other fields go about building these large data sets. And so we can think about the different carrots versus sticks. If you're trying to get a donkey to go somewhere, you either entice it with a carrot or hit it with a stick. In order to do it, look at other fields. So in the case of modern biodiversity, there's been a big carrot in the sense that a lot of funders have really wanted this to happen and pumped a lot of money into it. And museums have largely taken over this task of assembling the biodiversity data. Museums are also great in this respect. They don't have this publish or perish mindset that we have in academia. And so they have the kind of time and space to assemble this large data set. So that carrot works really well. You can also think about kind of a hard stick approach. I realize this might be a little hard to read from the back of the room. This is when I had a kind of 5-year sojourn as a molecular biologist, and I sequenced genes from a number of different animal groups, trying to understand when different animal groups evolved in geological time. What you see in this paper in the materials and methods is that new sequences are submitted to GenBank under these exception rules. And the way it works in molecular biology is that journal editors will absolutely not allow you to publish unless you have put your data into the accepted central repository with all the required metadata for those samples. And so that works really well because everyone needs to publish and you can't publish without it. In contrast, I'd say that the geosciences has taken maybe not a carrot or a stick, but maybe a kind of wet noodle approach to getting people to accession their data. So how many people in this room have collected a geological sample? Yeah. So most people, right? How many people in this room have gotten an International Geosample Number, or IGSN, for their sample? Nobody. Yeah. AGU made a big deal of the fact that they strongly encourage people to get IGSNs for their sample. So they're encouraging this, but there's actually no carrot or stick behind it. It's just a suggestion. Funders are also increasingly requiring people to put their data into central databases. But this kind of, which is I said, a good thing, but it kind of falls down for a number of reasons. The first is that a lot of my research, at least, I don't know about other people, is not actually funded by our central funding agencies in the US. So these are all the places I've worked, and only very recently have I received money from the National Science Foundation, which requires this data accession in order to do my work in West Canada. Further, even if people are accessioning their data or making their data available, this could be very different things. So the first thing is that there's a bunch of different repositories. So people might put it in EarthCamp, they might put it in Pangea, Zenodo, they might put it in a supplementary data file associated with this paper, so it's not aggregated anywhere. Even worse, sometimes people put these as truly horrendous as a Word file or a PDF, so the data is almost entirely unusable. There's also no requirement to provide any specific metadata like age or latitude or longitude, ecology or anything. So people are doing a better job of getting their data out there, but it's not in any sort of centralized way. So I think we can start to sum up where we are in the state of the field. So will sedimentary geochemists reposit their data because it's encouraged? No. Are the funding mandates helpful? I think they are helpful, but they're not solving our fundamental problem. Will people go out of their way to accession data that they published 5 or 10 years ago and is not under any sort of mandate? Clearly not. And so we need some sort of carrot that is going to get people kind of off the couch and get their data out of Excel files and out of their geological notebooks and into a single repository. And so what we kind of hit upon is the model that's been developed in biomedical sciences, which is a research consortium. The basic logic of this is that people want to be involved in cool science and cool papers, and they want to be part of an intellectual community. And the end result, a side product of this, is that all of the data is then available for people to use and reuse down the line. So I'm particularly familiar with this through the work of my wife, who is also a professor at Stanford. She works on the genetics of complex psychiatric diseases like schizophrenia, and I'll show a couple of slides because it's, I think, instructive about where this research consortium model can go in the long term. So this consortium has been around for 15 or 20 years, involves researchers from 38 countries, now has genomic data for millions of individuals, all kind of working together to answer these questions of the genetic basis of psychiatric diseases. And so this is a paper that my wife currently has in review, and she did a nice. Oh, it's not. Okay, well that's library. All right, so she did a cool retrospective analysis of what the consortium was able to learn as they assembled these data sets. And so basically what this is the number of individuals where they had the genetic data for, and the number of genetic loci that they were able to unambiguously relate to schizophrenia. So back in 2009 or so, they had a Nature paper talking about the nature of schizophrenia. They had data from 3,000 individuals. They weren't actually able to identify any loci associated with schizophrenia. Now, what my wife actually wanted to do in this paper was look at what cell types in the human brain, so there's 416 cell types in the human brain, which of those are related to schizophrenia. And of course, because we have no genetic loci that are associated with schizophrenia, there's no cell types. Flash forward to the group's next paper, 22,000 individuals, we can start to identify 5 loci associated with schizophrenia. However, and these are the 416 cell types, in order for it to be associated with schizophrenia, statistically, it has to be above this black line. You're not able to identify any cell types. Move on, more than double the data set. You're now able to identify 108 loci and start to identify 63 cell types that are associated with schizophrenia, and so on and so forth. What you can do then now is look at where these cell types are occurring in the brain and actually understand the cell types and the locations that are actually affecting or causing schizophrenia. And this is the first time we've ever had any idea of where in the brain these genetic changes are causing this complex disease. And so this is hopefully then the first step towards treatment of drug effects. The important thing to remember here is that each individual genetic study is only 100, 50, 200 people. It's pretty small. So back when everyone was compiling their data together and they only had a small population, they actually weren't able to tell anything. It really took the entire field coming together and bringing their entire data sets together before they could make these emergent properties come from the data set. And so to me, as a geologist or a stratigrapher, I think this is a really good analogy. I feel like we're each looking at our individual snapshots of Earth history. I'm looking at the Red River group. You guys are looking at the Bamboo group. You're looking at the Dos Chanteau. And it's only by combining all of these data, these snapshots together, that we can start to understand these trends through Earth history. So in order to do this, we started this community consortium, SGP, obviously just stealing this idea from my wife's research. You can read here, a community-oriented research consortium that seeks to address questions of environmental evolution across Earth history through statistical analyses of the sedimentary geochemical record. I'll talk a little bit more about this. There's 2 parts. There's getting the data set together, and then there's actually analyzing the data set. I'd encourage you, if you'd like to be involved, and it is open to everyone, everyone who would like to contribute data and be part of the consortium. We have both a promotional website and our search website. We now have 110,000 samples in the data set, coming in on near 4 million results, geochemical results, being built by 165 collaborators worldwide. We do encourage everyone to be involved. For the most part, what people do is they help code that critical geological context data for their sample. So this is just our standardized context sheet where people are filling in, depositional environment, or what type of sample it is, and so forth. So then what's the actual carrot that we try to give people? One is authorship on papers. So this is basically intellectual reward for your work in getting this data into the central database and providing your geological metadata in an accessible format. Let's see if I can, the PDF cut off the other points. Let's see if I can remember the other reasons why you should join SGP by heart. The other is being part of a community, an intellectual community. So we have social hours and meetings at Goldschmidt and GSA where we buy you beer and things for helping put your project into the database. We also run series called proxy primers, which are helping people understand or get all on the same page about geochemical proxies. And then finally, long-term accession and utility for your data. We do have a website. This is our first data release, which came out in late 2020. Our next one is going to come out in early 2025. So currently you can access about 80,000 samples worth of data. In January 2025, that'll bump up to 115 or 120,000 samples, with much better sampling of the protozoa. The second thing, and this is what distinguishes us from I think some of the groups like EarthChem that have been trying to build big databases, is we're fundamentally not a big database project. We're fundamentally a group that's trying to understand Earth's evolution. And so as a large part of that, we've been trying to come together to better understand how to analyze these large geochemical data sets of time. Because I think we've been pretty good as a field of going out, collecting rocks, bringing them back, making measurements. We've been less good about thinking deeply about how to analyze these data sets in time. It was fun today talking with the folks from Rio de Janeiro about possible kind of next generation ways to do that. I just highlight here a paper that actually Mira published in GSA today, thinking about kind of spatially and temporally related bootstrap approaches to analyzing these data. So basically our large goal here is to build a community of practice for statistical analysis. Okay. All right, so with that big introduction to SGP, I'm going to move on to some of the research questions and kind of my main research question that I'm trying to be focused on over my career. And this is how oxygen levels have changed through geological time and how this has affected animal evolution. So this is a figure, it's kind of based on the one by Kump that we see ad nauseum in the kind of conferences that I go to, the history of atmospheric oxygen through time. So this is a log scale of atmospheric oxygen. And so you see during the early part of Earth's history, the Archean, we have really low levels of atmospheric oxygen. This jumps up then orders of magnitude during the Great Oxidation Event. The early, or kind of middle part of Earth's history is believed to be relatively low, maybe 1 to 10% of present atmospheric levels. And then we have this second rise in atmospheric oxygen, the neoproterozoic oxygenation event, which, Graham, not to put you on the spot, Graham's against one. Did you coin the neoproterozoic oxygenation event?
Yes.
Yes. So we have the coiner of the term in our midst. Yeah. And so it's this second rise to near modern levels that has canonically been pointed to as the causal trigger for the chemical explosion. It's worthwhile thinking every time you see a graph like this, of what is the data behind this graph? Because if we think of something like the GOE, we can actually think of the number of long-term records that strongly support this. So the lack of, or the disappearance of sulfur mass independent fractionation, the disappearance of detrital pyrite and uraninite, the appearance of red beds. There's a whole bunch of records that support this. In contrast, when we get to this record, it's difficult to find kind of one single record that really supports what we draw in the cartoon version. It's worthwhile now, I would suggest going back and reading this paper by Mills and Canfield in 2014. It provides a really good history of thought on this. And what we find is that a lot of the evidence for the origin of the neoproterozoic oxygenation event has historically been this simple fact that there's no animals back here, there's animals here. There was a hypothesis that this was caused by oxygenation, and then the fact that we had animals was kind of convoluted back around to actually provide evidence for it. So I guess the take home is that our geochemical evidence for this is a little more lacking than what we see at the GOE. Despite this, when I was a grad student, there were kind of 3 central papers that purported to show really strong evidence for the neoproterozoic oxygenation. And it's worthwhile kind of walking through this. The first was this paper by David Feit and colleagues in Nature in 2006, which has to do the Shurham carbon isotope excursion that was heard about. they called upon the oxidation of a large pool of dissolved organic carbon. As we just heard, whatever's causing this large negative carbon isotope excursion is a really interesting question. I think the questions that have come up about this paper as heralding the neoproterozoic oxygenation event, there's been a lot of questions about to what extent carbon isotopes are necessarily faithfully recording the carbon cycle. We can argue about that. There's also kind of, I'd say, a lack of evidence for long-term redox change in this time. So we heard about how there is evidence for oxygenation at that time, but then if you look in the later ED acrid, there's clearly evidence for very reducing conditions. And so I think there's this trend where every time someone saw an oxygenation event in the neoproterozoic, they said that's the neoproterozoic oxygenation event. But what's really happening is that oxygen's going up and down the wall. So next was this paper by Canfield and colleagues in 2007 in Science. They used the iron speciation proxy to argue for deep water oxygenation at 582 million years ago. I would argue that, first of all, this is a local signal, and I would say that looking back on this 15 years later or whatever, what the signal from that paper is a little unclear. But also clearly global iron compilations show a more complicated story. And in fact, in Canfield's next science paper in 2008, he published another paper from the same time, or another section from the same time period, which showed exactly the opposite trend. So a complicated story to be sure. And then finally, in 2008, Scott et al. Published this paper in Nature looking at the molybdenum contents in euxinic shales, which they saw an increase in around 550 million years ago. This was then followed up on by some nice papers from Graham's lab and others. I would argue that this is still the best record. These records are still the best long-term record we have of the neoproterozoic oxidation event. The issues with these studies as they stood at the time, they had relatively low sampling resolution for time. So in fact, a lot of these peaks are basically kind of the late ED acrid and then the Devonian. It looks like there's good sampling, but remember that's 2.5 billion years of Earth history. These studies are typically not accounting for confounding variables like total organic carbon or aluminum contents that you would like to. And there's a focus on the highest points that are observable rather than the data's central tendency. And so, because this is what I'm going to spend the rest of the talk talking about, I want to dig into what this record actually is. So again, this is going from 2.5 billion years ago through to present. You see very low levels here, and then an obvious increase there. So the question is, what does this actually represent? So this is a schematic of how you interpret these records. I'll go through it and then try to provide a simple take-home of how to interpret the records. So this top panel is a modern, well-oxygenated ocean. So we take these redox-sensitive trace elements, things like molybdenum and uranium, and they're primarily delivered to the oceans through the rivers. Now, the main way that they exit the ocean is through reducing sediments, although the exact conditions required for each element to be sequestered into the sediment are going to differ by element. So basically, we have elements coming in. In the modern ocean, though, there's only a couple of little places that are actually anoxic or sulfitic. And so what this means, you can imagine you have like a big 5-gallon bucket with a hose coming in, and you've only punched one little exit hole. And so what that means is the water level, or in this case the redox sensitive element reservoir, is going to grow up really high within that ocean. Now, it turns out that the amount of redox sensitive elements that you get in any specific anoxic place is going to be related to the size of your dissolved redox sensitive element reservoir. And so if you have really high redox sensitive element reservoir in this well-oxygenated ocean, you're going to end up with high sediment concentrations. In contrast, you can imagine an ancient, poorly oxygenated ocean. Now you have anoxia or reducing conditions everywhere. If you have the same flux of redox sensitive elements coming in, now your bucket, you've taken it and you've just like jammed 100 holes in it. So any water that's coming in is essentially going to go straight out, or any redox-sensitive elements that are coming in are going to get sucked straight out into the sediments. And so your dissolved reservoir in the oceans is going to end up as really low. And because the amount that you're getting in any given reducing place is going to be related to the size of that dissolved reservoir, you're going to end up with a low sedimentary concentration. And so kind of, if you want to ignore all of that elemental cycling, the take home for this is that in a well-oxygenated ocean with relatively few reducing sites, you're going to end up with high redox-sensitive element concentrations. And likewise, in an ancient poorly oxygenated ocean, you're going to end up with low sedimentary concentrations. And so that is essentially what Scott and all of Christ have seen. Really low levels going through here, and then rising up about 550 million years. In fact, they'll show until number 4. So this is what we wanted to address using our expanded compilation from the SGP database. This is work that was led by Richard Stockey. He was formerly a Stanford grad student. He's now a professor at Southampton. And a large number of people whose names are too small to see here, but who are all involved in the data compilation and are authors on the paper. So our goal here was to be able to utilize and leverage all of that geological and geochemical metadata that we have in the SGP database. Or in other words, we want to take a multivariate approach to understanding how these redox-sensitive elements concentrations have changed over time. The way that we do this is a relatively straightforward machine learning method that's called random forest. This is based on regression trees or decision trees that you might be relatively familiar with. However, these trees can be relatively prone to overfitting. And so what you do is instead of using one single best tree, you actually grow a forest of trees. Each tree is going to use a bootstrapped collection of the data, and at each node or each decision point, you're going to use a random subset of your predictor variables. So in our case, that's things like TOC or basin type or morphology or aluminum confidence. So you're going to pick a random collection of those variables at each individual node. The end result of this is that maybe each tree is not as good as your perfect one, but because you're kind of building all these collections of trees, it's a little bit like wisdom of the masses, and you're gonna get around this problem of fitting. And this technique gives us two plots that we're interested in. The first and main one that I'm going to show here are called partial dependence plots. And basically, this is showing the marginal effect of a predictor that we're interested in on our response rate. And in particular, the main thing that we're interested in as paleoscientists is the marginal effect of geological time. Or in other words, how do molybdenum concentrations change through time when all of our other geological and geochemical predictors are constant? That's what we want to try to answer. The second kind of plot that this can give us is called variable importance plots. I'm not going to get too much into these. This is basically rerunning the analysis, but randomly removing the variables for each of the predictor variables in turn, and looking at how much worse the model gets. The take-home message for this is that this is basically increase in model misfit or increase in mean squared error. So higher numbers mean it has more impact on the performance of the model. You see things like total organic carbon and geological age have an important point. But the other take-home message to this is that all of these other things, so site type, metamorphic den, etc., have a positive influence on the model. And so all of the geological context that the stratigraphers and the geologists provide to us does have an impact on the model. All right. So let's dive into the data. I'd like you to focus on these two plots here. We'll talk about the other plots in a bit. These are these partial dependence plots for molybdenum and uranium going from time through 1 billion years ago through the end of the Carboniferous at 300. And this is the Ediacaran Cambrian. So this is where those previous studies saw that big increase in redox sensitive trace metals. And you see that when we account for all the variables in the model, all the new data that's been presented, we see a very different story. So we see relatively static levels going through the Precambrian. I would note that these methods are going to smooth out a lot of fluctuations that likely could exist in the Precambrian. We see a little bump up around the EAC and Cambrian boundary, but then it kind of goes down within uncertainty through the Ordovician and Silurian, where those previous studies didn't have good samples. And then really the big jump is sometime into the Devonian and Carboniferous. The other thing that I'd like to point out, because we return to it later, this is total organic carbon in shales, goes along, actually does experience a bit of a stepwise bump around the EAC and Cambrian boundary, and then keeps on going up through the rest of the Paleozoic. So basically, Rich summarized this as saying there's relatively low Neoproterozoic molybdenum and uranium in seawater, a moderate transient increase in the early Paleozoic, potentially a relatively large one around the earliest Cambrian, we'll talk about later, and then the major one being in the late Paleozoic. So the question now is, what does all this mean in terms of numbers that we can actually relate back to early animal evolution? This is where it really gets into Rich's field. I'm sorry, I'm going to rush through this in the interest of time. There is a preprint of this paper that's available on Research Square, and Rich should really be the one telling this story. So I'm not going to do it justice, but I will try to walk through this to get to the results more fast. So what Rich tried to do here was take an ensemble Earth system modeling approach. So he used the CGE model, and he ran it against a number of different levels of marine phosphate, going from 1% of moderate to 100% of moderate, and a bunch of different levels of atmospheric oxygen, going from 1% oxygen to 100% oxygen. And these are maps of the bottom water oxygen concentrations as oxic, suboxic, or anoxic bends across this model space. And so the results are fairly intuitive. If you have low oxygen and low productivity, you end up with kind of broadly suboxic bottom water conditions. If you have modern conditions, you have modern oxic levels of seafloor. Rich then only considered the configurations of oxygen and phosphorus that are stable on geological timescales. This is from Devin Cole's CANOPS model. So that reduces our space to something like this. He was then able to make maps of the bottom water oxygen conditions, basically looking at increasing oxygen and increasing phosphorus. And this is the fraction of the seafloor that's anoxic, the fraction of the seafloor that's suboxic. And you see that they're both kind of increasing as we increase oxygen and phosphorus. This is the step that I'm really going to blow through, just because I apologize for that. Rich basically coupled these seafloor maps to a trace metal mass balance model and the CANOPS model for organic carbon burial, and this is basically what he found. And the specific take-home that he found is that trace metal concentrations and organic carbon burials have different sensitivities to oxygen and phosphorus. So specifically, if you look at our trace metals, this is molybdenum and uranium. Again, this is increasing atmospheric oxygen. You see that the trace metal concentrations don't really change much across a pretty broad swath of relatively low oxygen levels. And this is something we've kind of always known about these trace metal systems, is that they don't respond very well at really low levels of atmospheric oxygen. In contrast, organic carbon burial increases in a much more linear fashion as you're increasing oxygen and phosphorus. And so basically how Rich interpreted these three stages of the data are a first stage where you basically have low total organic carbon, low trace metals, low atmospheric oxygen, and low productivity. So you're in kind of this phase. You then increase slightly as you move across the Precambrian and Cambrian boundary. You have higher TOC, but you still have relatively low trace levels, those blue colors. But you are kind of moderately increasing atmospheric oxygen and conductivity. And then you have the final jump in the Paleozoic up to high total organic carbon and seawater trace metal contents. So if we sum that all up, we end up with something that looks like this. So this is basically, again, a time scale from the Newtonian through the Carboniferous, the Ediacaran Cambrian boundary, the Cambrian explosion right there, suggesting that as we move from the Neoproterozoic into the early Paleozoic, we're probably increasing atmospheric oxygen, but just a bit, perhaps 1 to 10% of atmospheric oxygen to something like 10 to 40 or 6 to 40% atmospheric oxygen. I wouldn't say this is a particularly new insight. A number of people have suggested this, but I think this is the best evidence that there was a small but present increase in oxygen as we moved across the Ediacaran Cambrian boundary. So the question then is, does this matter, or how does this matter? So we're not seeing this increase to 100% atmospheric oxygen that everyone always shows in their cartoons and calls upon to drive the Ediacaran Cambrian transition. What's really interesting. So this is worthwhile to see. So this is a ROV footage from the San Diego margin at about 530 meters water depth, 15 micromolar O2, or about 6% of surface ocean levels. Might be a little grainy and hard to see here, but I'll try to point it out. So that's a crab, a sea urchin, that's a sea pen, that's a sponge, that's a brittle star. I don't know what that is. There's like little brittle stars sticking out here. That's a little fish hanging back out there. Another sponge. Another fish. Some more sea urchins. You probably get the point. This is a fauna with, kind of calcareous things sitting there in relatively high abundance, relatively high diversity. It's not that different from the Cambrian explosion. So we don't actually need 100% present atmospheric oxygen in order to have oxidation drive the Cambrian explosion. All we have to do is cross that critical ecological threshold for what animals require. Yeah. So basically the take-home message from, I think, all this work is that there's little evidence for the canonical Neoproterozoic oxygenation event in the way that we draw it on our figures. But a small change could have still driven a really large ecological response. I'm going to put this quote here that Rich wrote at the end of the paper, which I think is really good. We therefore argue that whether there was a Neoproterozoic oxygenation event depends on spatial and temporal perspective. From a global oceanographic viewpoint encompassing the deep ocean, there was essentially no NOE. From an ecophysiological perspective, considering that shallow shelf environments are where most animals live and most of our fossil record is preserved, there was an NOE. So basically, this leaves us kind of at a point where it looks like this oxygenation event is probably in the right magnitude to have been involved in the Cambrian explosion, but it's still difficult to say that it was necessarily the cause of trigger. So last few seconds, future directions. These are basically, I think, maybe I spent too much time at Harvard and Cambridge, Massachusetts, where everything in early animal evolution is driven by oxygen. We've spent decades thinking about oxygen, and I think we've neglected the role of other ecophysiological and metabolic drivers for animal evolution. So this is a figure that Rich Stockton and I published a few years ago which is kind of qualitatively showing that animal ecosystem responses to low oxygen or low food are basically the same. And so we talked about how oxygen is increasing and productivity is also increasing. The chief chemists always talk about productivity as a control on oxygen. But productivity also really matters for animals, right? It's what drives the base of the food. The second thing is temperature. And I feel guilty about saying the temperature is important because for a biologist in the room, that's about the dumbest statement you could ever make. Nonetheless, I think we have really been ignoring the role of temperature too much. These are some analyses, some respirometry analyses that my lab has made, particularly my former postdoc, Murray Duncan, who's now an assistant professor at the University of Seychelles. And this is the minimum oxygen requirement that's required to maintain standard metabolic rate for this purple CRT versus temperature. And what you see is that that minimum metabolic rate involves an exponential increase in required oxygen as you move through temperature. And so what this means is if you're only looking at oxygen, you're not getting the whole story. Oxygen and temperature are synergistically connected, and we have to consider both of them together. Likewise, if you're only considering temperature and you're not considering oxygen, you're only getting part of the story. And so I think that having kind of a more holistic view of oxygen and temperature together will really give us a much better understanding of our so, thank you. It's kind of cool. It's not. It's just a view from the helicopter looking down in this section. So, and there's rocks. And so this is the lower order vision. So you can just like keep walking basically all the way up to the front. Sorry, real camping. Spend too much time in the middle of the night.
It's water is economic always on the boat around.
There's like right around this corner, there's a class 6 waterfall. But I think no one has ever left. So yeah, you could try.
So this otherwise people online won't be able to hear. But you have questions for Eric? It was a very interesting talk, and I have lots of questions, but just open for more young here.
Just a short question, and I know you in past years you did a lot of approach to try to understand the influence of oxidation, LOE. And you talk now, this talk given a lot of trace element metals. Okay, database issues and really evidence for the LOE. And otherwise, there are a lot of isotope chemical data issuing some evidence, even these not really quantified content of the oxygen, both in the atmosphere and the ocean. But here showing something in chemical transition is something happened, even not 100% PA, but they are showing great change in that interval, even maybe later time to the Paleozoic, what do you put? Somebody the Paleozoic oceanic event, maybe that level maybe even higher than the chemical transition interval. So my question is whether you considering the other, whether the trace metal, the trace element metal, and also isotope data can tell you some different story, whether in the future you try to do similar analysis to use the Earth system modeling to understand why the trace metals show such kind of trend through 1 billion years, since 1 billion years to now, present days. Okay, this is one question. Second question is, so you are trying to convince people say oxygen or other temperature maybe is not really equal physical constraint for the animal evolution, like the chemical explosion. Whether you can expect any other reason maybe responsible for the chemical explosion. And really this reason deep ocean animal things will issue in the oxygen ecology, the ecological world there, there are a lot of animals living there. But if you consider Cambrian version, a lot of large movable predators there in the early Cambrian. As only that kind of animals or complex species may need more oxygen than what I need more oxygen. So this may be my question. Whatever you.
Yeah, that's a lot of great questions, and I'll try to keep them in my head. So with respect to isotopes, I think isotope studies are great. They are. I look forward to applying this kind of approach in the future. In some cases, the data density, at least when we started the project in 2015, we're not there, and so it's hard to build that long-term record, especially as you get into the Paleozoic, which I think is really the key. Is not just having the Ediacaran, but also having the Ordovician and the Silurian and so forth. The data records were not there, in part because as people go into the Paleozoic, they tend to just focus on these events. And so that's one of the reasons why we did this peer-reviewed study, is we wanted to look at something across 121 years. So yeah, I think I look forward to. I think isotopes are great. We've been working with people on thallium isotopes. Yeah, to have this long-term, to have the really long-term record. I think we're getting there. So yeah, I think they're great. I don't want to downplay them. The other thing I would mention is that I do think there's a possibility that in the earliest Cambrian, you do have quite high levels of atmospheric oxygen. I think even if I want to avoid replacing one cartoon version of something with another cartoon version that sticks around forever. So even if we talk about there's an increase in oxygen into the early Paleozoic, and then another oxygen increase in the later Paleozoic, it's still clear that there are fluctuations, and so there's nothing that prevents 100% PAL in the earliest Cambrian. With respect to the
Oxygen temperature.
yeah, with respect to the oxygen requirements of the early Cambrian biota, I think that's a good question, an open question. I haven't been convinced that there's anything there that has actually really high energetic requirements. You could push that number around a little bit, but if you look in the modern ocean, most things actually don't care that much until you get down to actually less than 10% of surface ocean levels. Most things that are adapted to those levels. The counter argument to that, which is really interesting, is that all of our modern oxygen and low oxygen analogs, basically, are oxygen minimum zones, which are also cold and don't change temperature. And so there's questions about how good those analogs actually are, or how much oxygen you actually need if it's 30 degrees Celsius in the surface ocean. And that's where I feel like we have basically no idea. Other questions? Yeah.
What proxy will you use to measure
That's a great question. For those online, the question is, what proxy do we use to indicate temperature? And this gets out of my realm. People have Kristen Bergman at MIT has made big compilations of oxygen isotopes through time, trying to look at how temperature changes. There's diagenetic and other questions with those, so it's difficult. I don't have an answer. I do think we can start to think more qualitatively. I've been very impressed with some of the studies that have come out of Tom Hearing's studies, for instance, where they're using qualitative measures of climate to calibrate their system models. I don't know if the results are necessarily correct, but I think that kind of approach can be interesting, and that's one of the reasons why I've been interested in these reports of glacials at different times in the Precambrian. Is not necessarily because they tell you about stuff on Earth, but if you have a glaciation somewhere on Earth, that tells you that the tropical surface temperature probably wasn't 38 degrees Celsius, and that difference between 38 and something like we have today has really important implications for ecophysiology.
Yeah, we can continue this later. Right now, we just thank Eric again, and we're going to call Rachel from University College London to give the last talk of the day. Okay? We need to set up something, and then let's go.
Tá muito frio?
cycles. So in the 1980s, 1990s, there was an international group called the Precambrian Subcommission. They didn't have this record of zircon abundance, but they found the same tectonomagmatic cycles nevertheless. And we now know that what they mean, we think, that these are orogenic cycles. So where we have a peak, let's look at this massive peak in here, we have the coming together of Gondwana, coming together of supercontinents. So each peak kind of represents the amalgamation, an orogeny for a supercontinent to come together, and then the darker green is the tenure of the supercontinent that gradually gets penoplaned, and then it sort of splits open again. It breaks up in these red zones, and then it all starts over again. So cycle after cycle after cycle. And when we actually look then at our subdivisions, this is the Archean Proterozoic boundary. This is not the formally defined Paleoproterozoic, Mesoproterozoic, but it's the one that many people use. The Mesoproterozoic, Neoproterozoic boundary, the Precambrian, Cambrian boundary, and actually we can even include the era boundaries of the Phanerozoic. They actually all fit in precisely the same part of these tectonomagmatic cycles. After a zircon peak, abundance peak, so after one of these supermountain collisional orogeny events, erosion events essentially, And they are expressed not just in tectonics, but they'll also be expressed as the formation of new basins. That after erosion, you'll get renewed sedimentation. So this was recognized way back when they actually came up with these subdivisions in the first place. So I'm going to explain a little bit about where these come from, and where this will take us in the future. And then I'm going to have a look at some of the things that we've been talking about, so case studies of events that we can see in the geological record and how they relate to these tectonic magmatic cycles. That essentially you repeat events again and again and again, but the Earth system is evolving. So those events have different expressions on the Earth system, have different consequences. So the first thing I'll look at is the Lomagundi event, and then I'll look at the Ediacaran-Cameron events, and I'll be focusing on the carbon isotope record. This event here is a positive carbon isotope anomaly, and this is several negative anomalies. And they relate very well to that tectonic context. Before I get there, have you ever thought why we need a geological timescale in the first place? Why we need an international geological timescale? I mean, in any country like Brazil, you do your geological mapping, you make your formations and your groups, and why do you need to correlate that to other continents? I mean, it's obviously to work out the nature and pace of Earth system evolution. It's very interesting scientifically, but it's also important for other things. It helps us to communicate. So we all have this notion of GSSP. The base of the Silurian period is an international GSSP, which means that if I am in the Silurian in China, I am basically in the same period of time that you're in Canada. Okay? So ease of communication. It helps us to make geological maps that are instantly recognizable. It helps us to reconstruct the pace and the nature of evolution. They help us in exploring resources through correlation. They're also an intuitive pedagogical device, and I'm going to talk a little bit about this. That for every period, it's important to know what is the concept of that period. Is it correct? Is it of global significance? And if possible, going forward, we can retain some of that context in the name so that we can immediately communicate not to just geologists, but to non-geologists as to what it actually means to be in the Cambrian period. Okay, for teaching, it's really important. So to talk about that a bit more, most periods therefore have a conceptual basis. So if I talk to a Russian about the Ordovician period or the Paleoproterozoic era, they will have a concept in their mind which is basically the same as my concept. I'm going to be talking about some of these periods. Cambrian period, which has a concept. It's the beginning of animal carnivory, of shells and burrows, and it's a diversification of animals, the Cambrian explosion. The Ediacaran period is the deglaciation from the snowball earth and the rise of animals. And the Cryogenian period is conceptually very strong. It's a time of tremendous cold. So these sorts of concepts were in people's minds when they came up with the Precambrian subdivisions of the Stenian, the Ecpasian, etc., which of course very few people use. But I'm hoping in the future when the concepts are clearer, we'll start to use these in a bit more detail, a bit more often. But these all have concepts. These are Greek names, okay? But they're not like Phanerozoic GSSPs, right? They are not in a rock body somewhere in the world defined by GSSPs. These are what's called GSSAs. So the boundary, for example, of, let's say, the Rhyatian or Assyrian is 2000, I think it's 2004 to 2050, okay? That's an exact age, a rounded number. So conceptually, it's not very strong. It's not rock-based. It's just, even though the concept is within the name, or Assyrian means mountain building, Rhyatian means rivers of lava. So there is some concept behind it, but the concept doesn't necessarily fit this GSSA, this exact, precise, absolute age. So going forward, I'm hoping that we can actually change some of these and make them much stronger on a global basis, so that we keep the same concept, and that concept is valid internationally. So in other words, to sum that up, I mean, we have quite a different way of subdividing the bits up here than we have the bits down here. and we're gradually working our way down to make these conceptually much stronger. But it's important to know that they all have concepts. Okay. So we've talked about these major boundaries, the Archean Proterozoic, the Paleoproterozoic, Mesoproterozoic, all of these boundaries which are relating to tectonic magmatic cycles, but they also have meanings in Greek. You know, the new Archean, the old early life, the middle early life, and they also have concepts as well. So anyway, I'm labouring a point there. But so to move quickly to how this is developing in terms of global stratigraphy. Names like the Cambrian, the Ordovician, Silurian, they were all set in the 19th century. Okay, there's a 19th century concept, but mostly by Europeans. Okay, and now we have set GSSPs, usually based on the appearance of fossil assemblages. We need totally different ways of doing this in the Precambrian, and this process started of GSSP creation already in about 2004, when the Ediacaran was the base of the Ediacaran was defined. Okay, and so that when you are this is Australia, the Nuckalina Formation in here, which is a cap carbonate, and this is below it the Alatna diamictite, so evidence, striated, faceted, polished stones in this red diamictite here. So this is conceptually the Cryogenian period, and then the deglaciation and the Ediacaran period starts right at that point. So there's no basis in fossils for this GSSP, but there is a sort of a basis in what we would call chemoceanography, this carbonate event, which is a worldwide phenomenon, and you can find it also in the Van Puy group at the base of the Settimagos. So this is quite recent. This is 2004. The people started to move down from the base of the Cambrian to the base of the Ediacaran, and then more recently we've been looking at the base of the Cryogenian. And a number of people, for example, Walter Bleger here, was the first to make a proposal to have GSSPs all the way down through the whole of the Precambrian, but that hasn't been done yet, but I'm sure it will happen at some point. So the GSSP approach is the way forward for chronostratigraphic subdivisions of Precambrian, but we're not there yet. So gradually we'll get rid of all of these GSSAs, replace them with GSSPs, so that we can do all of those things that I said a geological timescale is good for. We can do that also in the Precambrian. So chronostratigraphic subdivision begins with an agreed concept. Well, the concept of the Cryogenian, which was the next one to be considered, is basically snowball Earth, is glaciation. So the Neoproterozoic Subcommission established a concept in 2009 that it's simply related to glaciogenic deposits. It's all because Cryogenian means cold, so the Cryogenian period should begin when it starts getting cold. The problem was that the original GSSA was at 850. This is a problem with most of the GSAs, all the way down the Precambrian. And so then, very quickly, within the course of a year, it shot up to that point. So as soon as you change the concept of a subdivision, a time unit, right, you can then start moving it up and down the geological timescale. It's not a fixed rounded number anymore. It suddenly becomes a concept that you can apply all around the world. You can say, oh, in my country, this is where it is, and in that country, that's where it is, mythologically. And then at some point, after lots of discussion, you work out a level which is equivalent in all of those different countries. So it's somewhere around 17, 720 million, something like that. And that's a global definition. But it's still awaiting, of course, a fully defined GSSP. There's lots of discussions going on. Yeah, so that was ratified in 2014. So it's what we would call a GSSP pending. And there's lots of ways that you can actually correlate that stratigraphically, and you may be well familiar with this. We've had a lot of this kind of geochemical data so far. You can use things like carbon isotope anomalies in here. So this is the immediately pre-glacial negative anomaly in Scotland, the Garvalac anomaly, and there are also other anomalies that perhaps we could use for correlation at the finer scale as you go down the rock record. But that's essentially the cutting edge of stratigraphic correlation right now. There's nothing really below this for about 2 billion years that is globally agreed. Okay. So this is not totally a new thing to change GSSAs into GSSPs. If you read the Geological Timescale book 2012, an attempt was already made. Martin van Kranendonk and lots and lots of other authors suggested replacing GSSAs, see these rounded numbers, which are the clock symbol in here, and replacing them with concepts that could be found all around the world. But in doing that, unfortunately, we changed a lot of the names, changed the concepts, and that was very controversial because, as I pointed out with the zircon abundance record, actually the existing subdivisions are pretty good, and they are globally significant. So those tectonomagmatic cycles, those supercontinent cycles, they form the basic framework of the entire geological timescale, even into the Phanerozoic. So we don't want to get rid of that. So this hasn't been very popular and it's not been very used. So we tried to get a few people together who knew a thing or two about Precambrian stratigraphy, and we were invited to form a working group to see if we can have a slightly better job, but without actually removing a lot of those previous concepts. Try to sort of merge the GSSA and the GSSP concepts. So we submitted the white paper, which is still under discussion, but we published it in the meantime in JGS. You can see it's like a cast of thousands in here. I'll just take out a few names which may be familiar to you. I mean, we have people who were in the original subcommission who brought in the GSSAs in the late 80s, early 90s. So people like Reinhard's book, who is, I think he's in, where's Reinhard? Brasilia. So he was part of the original subcommission, as was Paul Hoffman, as was Kenneth Plum, who was the chair. So these guys are getting on a bit. It was quite difficult to actually find Kenneth Plum. He's been retired for like 20 years. But they were all very keen to come back and start working on this GSSP concept to replace the previous GSSA concept. And I guess there's quite a few other names that you can recognize here. The result of that then was our suggestion, which I showed you in the first slide, to come up with GSSPs. So that's what these are. These are golden spikes that you hammer into the rock, as people do in the Phanerozoic. See if we can actually come up with similar golden spike horizons right the way through the Precambrian, and keep that as conceptually strong as possible. Because once you have a concept, the age is changeable, and that's so key, right? We don't want an age which is fixed, because as soon as we get a new age, a better age, then we have to redefine everything. It cannot be defined on an age. It has to be defined on a concept. That concept defines a level. So these concepts are things like the GOE, the Great Oxygenation Event, and we suggest we should use the name Storium, which is the Greek word for rust for that period. And then Siderian becomes an Archean period named after iron, and that would incorporate a lot of the iron formations, for example, in Brazil. So this is still under discussion, especially the ones which are in italics. But this is going to be, I think, the future. I took a little bit longer over that than I thought I would, but I thought it might be interesting to see where we're heading in terms of the geological timescale. But there's no suggestion that we change all of these names because they all have some kind of interesting concept behind them. So the lava flows, we'll talk about that in a moment, is this first super mountain orogenic event. And then these are a bit similar. Stable, firm, cover, extension. And that's what we call the boring billion. So it's very difficult to distinguish things going on in that period. And we'll see where we get to with these. Okay. Right. So, take-home message there, don't throw the baby out with the bathwater. Let's keep the names. Let's reinforce the concepts and take it from there with a lot of discussion. Okay. So how are we going to do that in reality, those concepts? What concepts are we talking about? We can't just use tectonomagmatic concepts because you can't go to a sedimentary rock and find an orogeny, right? It's a global orogeny. It's just not going to happen. You can't use zircon abundance because that's a global record. It's not a local record. So you need things which can be found globally, and some of these things, one of the most important of these things is carbon isotopes, and it's really used as a metric for Earth system evolution. We often talk about the Lomagondi event and the Shuram, we've heard about already, but there are lots and lots of others, these carbon isotope events, and everything is built on that framework. So that's extremely important. So, but what do they mean? How do they fit into the tectonomagmatic cycle? So let's look at this one. This is the 2.2 to 2.04 Lomagondi Jatuli. It's a worldwide positive excursion. Right around the world, we find very elevated carbon isotope values of +5 to +50. Right? It seems to be a global occurrence. And interestingly, it fits just after the great oxidation event. So the atmosphere has become oxygenated, and then we start to find these high carbon isotope values. And that fits into the famous Lyons atmospheric oxygen curve here. It's this overshoot here. So it's a time of relatively high oxygen, but in any case, it's after the great oxidation event. We'll see what that means in a moment. How do we look at carbon isotope excursions? We've heard already a lot of skepticism about carbon isotope excursions. Are they diagenetic? Are they orogenic carbonate? What do they mean? Well, for me, I'm very conventional. And if you find very high carbon isotope values everywhere in the world for a particular time interval, that is telling us that there is an imbalance between carbon burial and carbon oxidation, right? You're burying carbon, organic carbon. And to understand that, we have to go back to people like Bob Gallons, the founders of Earth system science in the 1960s and 1970s. And they wrote these classic papers like coupling of the sedimentary sulfur carbon cycles. So they started from the principle that if we keep a balanced oxygen budget, okay, then we have to consider the two main sources and sinks of oxygen. So that is carbon and that is sulfur. So today, most oxygen in our atmosphere derives from carbon burial, but a very substantial part of that oxygen in the atmosphere actually derives from pyrite burial. So for every mole of sulfur that you bury as pyrite, as iron sulfur, you release 2 moles of oxygen, almost 2 moles. With carbon, it's just 1 mole. So sulfur, mole for mole, is actually more important than carbon. And when we're talking about an anoxic Precambrian world, sulfur would be considerably more important than carbon. So they call this the redox coupling between the sulfide, sulfate, carbonate, organic carbon relation. So essentially, if you're burying more organic carbon, which is what the Lomagondi event is kind of telling us, you have to compensate that, okay, with the sulfur cycle. So the reduced part of the sulfur cycle would be buried less. Because if you don't do that, what ends up happening is that you just have ridiculous amounts of oxygen in the atmosphere. That clearly didn't happen, or you end up in negative territory, which clearly didn't happen either. So you have to keep some kind of balance, and they realized this all the way back there. And if you ever have time, these are just fantastic papers, which are often published as book chapters. This is a book chapter. I think it's this one, which traces in the sea. So this is how it looks in a box model sense. So we're talking about this cycle in here of burial and weathering. This cycle of burial and weathering. This is the oxidized part, carbonate, which is being weathered, positive, and the sulfate, which is kind of doing the same. Okay, so you have the reduced part on the left-hand side and the oxidized part on the other side. So essentially, if you have an imbalance in this part, so you bury more organic carbon than you weather, then you have an excess oxygen flux. Now that excess oxygen flux will have to be compensated for by a decreased pyrite burial flux or an increased pyrite weathering flux. Okay, that's how it happens. Now, in reality, it doesn't happen like that. And that's because there is a major difference between carbon and sulfur. Carbon is what I would call a very tight system, both of these, in the sense that if there is an imbalance in carbon burial, and you produce oxygen, then you can weather more organic carbon. Likewise, if you have more CO2 in the atmosphere, you can weather more silicates. That's because it is not a quantitative process. It acts as a chemical buffer. Okay? That's totally different from sulfur. Sulfur is carbon's evil twin, right? Because if you expose sulfur to weathering, it will all weather. Because it's very easy to weather, it's very easy to oxidize. That's not the same with organic carbon. If we expose more organic carbon today to weathering, yes, some will weather, but probably not all of it. Even today, in our oxygenated atmosphere, let alone the Precambrian. So this can act as a buffer, but the sulfur system doesn't act as a buffer. So the only way to do this is to link it to the sulfur. So in other words, if you bury more organic burial, okay, and you've already weathered all of the pyrite that is available to be weathered, then what happens is the sulfate, which is the product of that weathering of pyrite, will have to leave the ocean as sulfate and not as sulfide. Okay. So sulfate burial, which is this bit here, as evaporates, is the sink of oxygen, which has been produced by organic burial. This is actually the classic way of explaining evaporite burial in the Permian, evaporite burial in the Cambrian, and actually the Lombardian ones as well. In mathematical terms, if A is greater than B, then C is greater than D. This is not isotopes here. This is just carbon and sulfur, and it has to happen to some extent like this. So what do we see with the Lombardi event? Lo and behold, we see that there's a sulfate-rich ocean during the Lombardi event. We have these very, very high carbon isotope values, and this is the very first time in Earth history that we get massive evaporated sulfate deposits. Hundreds and hundreds of meters of anhydrite and other evaporite minerals. So this is not just in this place, this is worldwide. I think this was a big shock when people drilled through this in Korea. Because we don't really think about evaporite sulfate in the Precambrian because we think there wasn't sufficient oxygen. But there clearly was here. You're essentially weathering pyrite, producing sulfate, and that sulfate is coming out of the ocean and it goes up into the ocean, and occasionally it comes out in these massive evaporite deposition events. Most of them are in the Phanerozoic because we have much higher sulfate, but occasionally we have these big events like the Tonian. There's a little one at 1.6, and then there's this huge one at 2 billion years. Okay. So the key point here, though, and it will come here, is if we look at this box model again, this is the modern carbon-sulfur system. If we go back to the GOE and the Lombardi event, etc., then the sulfur cycle didn't look like this, largely because we didn't have any evaporite to weather, right? Before the GOE, you couldn't even weather pyrite. So if we look at the pre-GOE carbon-sulfur redox balance, we have to balance everything without carbon weathering, without sulfide weathering, because we have detrital pyrite. That limits to a very large extent pyrite burial and sulfate concentration, and we certainly didn't have this cycle of evaporite weathering and deposition. There's no gypsum in the Archean, pretty much. It happens all after the GOE. And then at 2 billion years, it just becomes massive, very much like a modern-day evaporite giant basin. So once you have the GOE, of course, that changes. You suddenly do have pyrite weathering, sulfate builds up into the ocean, you can have euxinia at productive margins, you can have a limited amount of sulfate weathering, but actually not that much, because you haven't yet had any evaporite basins forming. So there's a lag time before you get to a full carbon-sulfur cycle. So what does that mean? That means when you weather pyrite sulfur after the GOE, and that will eventually lead to evaporite deposition, you cannot bring that oxidizing capacity back into the ocean through evaporite weathering, that there isn't any evaporite left to weather. So we have an incomplete cycle. And that may be part of the reason why the Lomagundry event is so extreme. Okay, so this is case one. These tectonomagmatic cycles are leading to a position of pyrite weathering is greater than pyrite burial. And that's a sink switch from carbon dioxide to sulfate. So the carbon dioxide is referring to the carbon burial. Okay, so you're taking carbon dioxide and you're making organic carbon and you're producing oxygen, and that's a sink switch from CO2 to SO4, and you have your evaporite weathering event. I'm going to go through that again if you follow those things. So you take the rock sulfide, you weather it, builds up sulfate in the ocean, and you end up with your evaporite deposition, and that's how you balance the oxygen budget in the post-GOE world. You have your excess organic carbon burial, which is your GOE to start with. That weathers the sulfide, and it comes out as sulfate. If you don't balance the budget, then oxygen will just go up and up and up and up, and it becomes nonsensical. So this is clearly somewhat what happened. There is another way to change carbon isotopes, and we published this way back when. I'm not going to go in great detail, but it also relates to tectonomagmatic cycles. So the Lomagundry event was a time of not uplifted erosion, rather the opposite. It was a time of a tectonic lull. So it's a time of a supercontinent being peneplated. So relatively low physical erosion, but relatively high chemical weathering. And that's because it was a time of high outgassing. If you remember, it was called the Rhaetian rivers of lava. So these are some of the biggest large igneous provinces that we know about. So that means that there's a kind of an imbalance between high chemical weathering, driven by the CO2 flux into the atmosphere, and then low physical erosion. So weathering intensity is very high. And under those conditions, you can actually generate a carbon isotope excursion, which is this one in here. Okay, this is burial, so I'm not looking at this one, it's this one. So this is the classic Lomagundry, but this is when you force it using erosion, so low erosion, high chemical weathering. You can also produce a carbon isotope excursion. So it's a combination of two that you need to explain the Lomagundry event. This is another way of looking at this, that the relative uplift rate is you can drive a positive carbon isotope excursion at relatively low uplift rates during a tectonic lull. So times of high carbon isotopes are times of supercontinent denudation. Okay, they're times of peneplain supercontinents, low erosion, but relatively high volcanic outgassing. And then you have to take into account the redox balance that I was just talking about. When you put all those things together, they are intimately related to tectonic magmatic cycles. Is there any evidence for this, apart from the geological evidence, that this is the time of a tectonic lull and high rates of outgassing? So this was a compilation that Ying and her student Helen did and was published a year ago. And you can see that the Lomagundry event, even though this is a preliminary compilation, fits into this little dip here in the seawater strontium isotope. This is the dotted curve, is the strontium isotope evolution curve. And if that's correct, then that would indicate either low erosion and or high rates of magmatism, because magmatic rocks have are relatively unradiogenic, and both of those things will drive a positive carbon isotope excursion based on what we just said. So there is an intimate connection between these tectonomagmatic cycles and carbon isotope. Let's look at the other way around now. So we've had a tectonic lull, we've had high outgassing. What about when you have these super mountain orogenies, uplift and erosion? What happens then? After the Lomagundry, we have evaporite basins. We have massive thicknesses, kilometer thickness of sulfate minerals, which eventually, in orogenies, will be uplifted and eroded. So there's a bit of a lag time. So it takes a long while till we get to the next major orogeny after the Lomagundry. The really big one would be then the formation of Rodinia, and then the formation of Gondwana is that super huge zircon abundance peak. So in those times, because it's a quantitative phenomenon, sulfate weathering, pyrite weathering, sulfate, that's quantitative. If you simply raise up a mountain, then all of that sulfite, all of that sulfate will end up in the ocean as dissolved sulfate, right? All of it, because it's a quantitative process. Because pyrite is very easy to oxidize and sulfate is very easy to dissolve. So if you do that and you think about the carbon cycle, you expose a lot of organic carbon, a lot of carbonate, it's not quite so straightforward in that case, because not all of the organic carbon will be weathered in that case. So you can predict that rather like a reverse Lomagandi, you will have times where there will be an imbalance, but in the other direction. So that you will have a massive flux of sulfate into the ocean, which brings in oxidizing capacity if you couple it with pyrite burial. Okay? And then what will that oxidizing capacity be used for? If you follow the Garrels and Werner ideas of sink switching, that will be coupled with carbon oxidation. Because as I said, carbon oxidation is not a quantitative process. Okay? If you have more oxygen availability, you have more time, you will oxidize more carbon. So that's not what happens with the sulfate. So I often say that these negative excursions, which are pretty amazing, if we didn't have them, I'd have to invent them, because sulfur cycle imbalance is just a natural side effect of tectonomagmatic cycle. And the biggest one we've already heard of here is the Shuram, but we can also talk about this, the Trezona, the Garbalak, we had a look at that earlier, Bitter Springs, and there are little ones all the way back here, but they don't reach this kind of size. But they clearly are important. They are massive perturbations to the carbon-sulfur exogenic system, and we can kind of see that because they have an intimate relationship to climate change in here. They all precede glaciation, even the Shuram. Okay, it's less well known, but the Lorchen glaciation in 560 follows directly from the Shuram. So these are events, if you look at it in a tectonomagmatic sense, these are events related to the coming together of Gondwana land, the supercontinent is smashing together what's often called the trans-Gondwana land supermountain, and just lots and lots of sulfate coming into the ocean. Nutrients as well, this is what Ing was talking about, and those nutrients drive pyrite burial, and pyrite is a very, very powerful oxidizing, has a very strong oxidizing potential. So how does that look on our box model? So we're going the exact opposite way. We start with rock sulfate sulfur. We end up putting that into the ocean. Sulfate rises in the ocean. And if you match that with pyrite burial, because you're bringing it into an anoxic ocean, which is nutrient rich, because of all of this orogenesis and erosion and weathering, and you bury it as pyrite sulfide, you release almost 2 moles of oxygen. Okay, it's a free lunch. It's an oxygen-free lunch because there's no need actually there's no kind of compensating mechanism here. This will just generate oxygen, and that oxygen can then be used to drive organic weathering, which is at least partly responsible for these negative anomalies. So each one, it may not be an oxygenation event. If I was to have my time again, I probably wouldn't call it the Neoproterozoic oxygenation event. I'd probably call it the Neoproterozoic oxygen production event or oxidation event, because what you're doing here is you're producing oxygen from sulfate burial, sulfate reduction, which is then used to oxidize organic carbon, and the net result of that is not necessarily anything. We don't necessarily have oxygenation as a result of that, or at least not massive oxygenation. But what you're doing is you're producing a lot of oxygen. So these are oxygen production events, which are equally important for understanding the exogenic system. This is a more cartoon way of looking at this. We've looked at this already. So sink switching from rock sulfate to net carbon oxidation, I would say that's a predictable consequence of tectonomagmatic cycles, and I would say that Lomagandi is also a predictable consequence of the GOE in the absence absence of evaporite weather. So what you're kind of doing here, sulfate weathering, sulfide burial, producing oxygen and carbon oxidation. What's very important here is the feedback, the positive feedback which goes back to chemical weathering and more nutrients. Because if you don't have the nutrients, right, you don't have the ions, you need an anoxic ocean rather than an oxic ocean. You can't bury the sulfide. So that will be self-limiting at some point. When you run out of sulfate, or you have an oxic ocean, or you run out of nutrients, that positive feedback will stop, and sulfate will just rise in the ocean. That's probably what it would do today. If we did this today, we had a super mountain, we weathered all this stuff, coming in, we wouldn't get a super sulfide burial event, because we have a completely different Earth system. So the Earth system context is extremely important when we think about the expression of tectonomagmatic cycles on Earth system evolution. Now this particular one, you've already seen this, so Marianne, I'm sorry you've probably seen this diagram many, many times. The reason I show that is not to show Marianne, but it's his work, is to show the intimate relationship between this oxidation event and biological evolution. So these things have a real world consequence. The state of the Earth system at that time was ripe for oxidation, and what needs oxygen? Animals need oxygen. And so there is some relationship definitely between this oxygen production, which can then balance the oxygen consumption that newly evolved animals require. Okay, it's not just how much oxygen do they need to live, they also need a production of oxygen to sustain their metabolism. Okay, so this is just, I put this in just to show you that I'm not talking complete rubbish about sulfate deposits. We still actually have 1 million kilometers cubed of Tonian evaporite today, all around the world. And how much there was originally before these erosion belts, I mean, it must have been an absolute, mega evaporite giant phase in the Tonian. So these sort of uplift and erosion events, they have to happen. Okay, and there are predictable consequences from them. It's just identifying when they happen. Okay. So, and it's intimately related to this cycle of rodinia supercontinent formation, supercontinent tenure, breaking up, and then smashing together to form a new mega continental supercontinent. So you need to take this tectonomagmatic context into account when you're thinking about carbon isotopes, sulfur isotopes, or biological evolution. And we often don't have this holistic, integrated approach, but I would recommend it because the geological timescale is simply based on that. This is Ing's work. I guess this is saying the same thing, except you can look at it with isotopes. So here's a strontium isotope record, which he constructed, and you can see that it matches quite well the large igneous provinces during the rift of rodinia. So it's sort of like this integration of different data sets, zircon abundance, isotopes, biological evolution, evaporites, put it all together, you have a much more holistic view of how these things came about. So that's sort of summarizing these two end members, but there's probably lots in the middle. So we have these times of continent tenure breakup, characterized by low relief, low erosion, volcanism, high chemical versus physical weathering. That's a very key controlling factor over things like nutrients. Carbon, I'm not going to go into this, but the net result of that is that you can then generate high carbon isotope values because you have high CO2 flux, but also have lower erosion. And you have the opposite with continental uplift and erosion, elevated riverine sulfate delivery, nutrients, eutrophy, and then high physical versus chemical weathering, which is what we have today. And as a result of that, you can generate negative carbon isotope. You can actually do this in the Cenozoic, but I'm not going to bore you by showing you this, but they're much more muted. So, to summarize all of that, tectonic magmatic supercontinents, outgassing erosion cycles form the framework to rock-based subdivision. You know, that's really the backbone behind everything. We start, so that's why I think we shouldn't throw the baby out with the bathwater. Those names, those subdivisions, even those ages, they actually mean something, and we should retain them if we possibly can. And they cause predictable changes for things like chemical weathering, physical erosion, and also the related coupling, the sync switching between the carbon and the sulfur cycles. I think that's probably all I need to say, and I'm probably overrunning hugely.
Thank you, Graham, for a very nice talk and kind of putting everything together. Yeah. So do you have questions for Graham? I have a question. You showed us a carbon residue population from your book, right? In 2023. Did you add the middle bamboo exclusion there? It looks like pretty high values before just after the Marigella there, before the skiers. I don't know if you added it.
So that would have originally been from the geological timescale. So it depends what they do, whether they put the boundary in that did. But there are very high values in other places too, not quite as space. That might yeah, which is also slightly anomalous. So there may be something to it.
Yeah, there was some hand at the back there. Oh, okay. Right. Sorry about that.
So, any other questions for Graham? Okay. Thank you. We also want to thank the people that organized this logistics for us. Mariana, Suzana e Matheus, todos o pessoal que organizou pra gente vir. E a gente vai tirar uma foto, né, com os palestrantes, com vocês também. So we'll make a group photo here before we leave. Eric, Inga, Mariam, Sean, everyone that's